traditional way of assembling. Now, for the brief introduction of the Professor Jang, I would like to invite the Dean of the Department of School of Basic Science and Faculty of the Department of Botany, just Professor Sanjeev.
Very good morning, one and all. Honorable Vice Chancellor, Professor Raghavendra P. Tiwari ji, Dean in Charge Academics, and special guest, Professor Jian. Well, it's a fortune for us that we have an acclaimed scientist who is basically a pharmacogonist, and then he has spent a significant time in US after his PhD, and then he's serving his own country as a foreigner because he's now a US national. So, but he's treated as a Chinese there because he's born and brought up there. So Professor Jian is a pharmacogonist, and he did his PhD from China. Then he moved to US, and he worked there in different capacities as a scientist, as a postdoc, and worked for almost 12 years, I guess, if I'm correct. And in 2000, he shifted to China back as a professor. And from last 24 years, he's last 14 years, he's working as a professor. So he has a vast experience of more than like 25 years in the field of metabolomics. He's a dynamic scientist with more than 118 high impact publications, which are published, if you're a plant scientist, published in like plant physiology, plant cell, etc. He has many patents. Patents being outnumbered. I couldn't count those patents, but since Vinay has written this 10, I guess it is more than 10 patents you have already in your account. As far as grants are concerned, he has executed millions of dollars of grants till now, and the highest was like 2000. It's like $20 million grant. So he's basically the current affiliation of Dr. Jian is Yunnan Agricultural University, China, and his expertise lies in metabolomics. So in coming 5 days, in coming from today to 3rd of December, he'll be talking about metabolomic techniques, various methods, extraction protocols, about the data interpretation, how to generate data, how to interpret data, how to normalize data, and he'll be supported by our own course coordinator, Dr. Vinay Yadav. So it's a fortune for us, and we, on behalf of Department of Botany, from School of Basic Sciences, and from the Central University, we welcome you, sir, to this Gyan program. And we feel that this collaboration, this whole thing will not be limited to another 5 days, but we'll have future collaborations also. And we'll have many more programs and research tie-ups in future also. So we welcome you once again. Thank you for sparing your valuable time and coming to India. I'm sure that you'll be having a good time here. As you mentioned that you enjoyed last night and traveled from Delhi to Punjab. So in another 5 days, we'll take you around also. Punjab is a very, happening or hot state as far as culture is concerned, as a diversity is concerned, and especially food is concerned. So once again, welcome you, sir, and welcome you all, the participants who have chosen this program. This is, it's kind of a mutual exchange. You're also expert, he's also expert, so there should be a, dialogue, not like a monologue. So I'm sure you'll be benefited with this program. Thank you all. Thank you very much.
Yeah, thanks, Professor Sanjeev, for the brief introduction. Now I would like to request the Professor Ramakrishna sir for filing this inauguration section, because we have the live classes at the 11 a.m. So because the short period of the time, we would like to bind up this program within the 10 minutes. Professor Ramakrishna sir, tell us live to the Gyan participants on the, because this Gyan lecture is live all over the India. So Professor Ramakrishna sir updates about the university, different programs, and a little bit about the departments also. And yes, one more important things that I would like to announce, that it's very difficult to get the patents in the biological sciences. We are facing a lot of the issues. So Professor Jain is the expert, so we try to understand how we can submit the good patents also from our departments and the biological sciences also. So yes, Professor Ramakrishna sir. Ramakrishna sir is the Dean Academic in charge, so he will explain the different academic activities of our university to the Gyan listener, whoever is sitting anywhere in the India and abroad also.
So good morning to all participants, Honorable Vice Chancellor and Dr. Vinay for getting the first Gyan program to the university. We have been trying, I think, for quite some time, but this is the first one successful. And Dr. Mukesh Yadav being coordinator. So when he took over coordinator, after that we are getting, and I think there is one more we got Gyan, and a few more are there in pipeline. Hopefully we'll get more of those. So to the participants who are coming from outside the university, so as you all know, we have 31 departments here. 16 are science departments, remaining 15 are humanities and social sciences. So we have a number of programs here, some of them undergraduate for 5 years started recently. We have 44 master's program and a number of PhD programs. So coming to, there are a lot of opportunities in the university, and we have made a lot of progress in the last 15 years. From the time we started as a small university with a few faculty. So Dr. Sanjeev Thakur, who is the dean of the school, has been here for a long time. So he knows all the history, what happened from where we started. I joined later in 2015. So this is now a really happening place, except for the last few years. Because of the COVID and all, we had to slow down. But now I think we are picking up, and we got a lot of projects. We are developing a lot of infrastructure, and research is going on also at a higher level. So we got a lot of projects like national level, several crores of rupees, and our faculty are publishing in very good international journals. So one of the things is Professor Zhao is here, so as he is an expert in plant metabolomics. So as you know, plant metabolomics is a frontier area in plant biology. So those of you who are here to learn about it, so as a student, if you are a student, we all hesitate first to go into a new area because we don't know what is the protocol, how to do it. So this is a great experience for you, opportunity rather, to learn the protocol, the principle behind it, and how to use it in your research. Make experimental design where you can incorporate. So you may think that we need facility like GCMS, LCMS or NMR. We don't have that. But you don't need that in your institution. Nowadays, so many service centers, facilities are there all over India. You can, if you know how to make the sample preparation and what is your plan, you make that one. And then send the sample out, including our own university. We have here GCMS and NMR. We are going to buy LCMS also in the next few months. So we offer that service. If it's an academic institution, you get it at a less at a good affordable price compared to other institutions. And so you can also collaborate with our faculty then and write projects, and that is one possibility. And Dr. Zhao is here, so he will give a lot of information regarding all the procedure, theory, the plant metabolomics. And as per Vice Chancellor, science is right to any or any, it's international. It's good to it and read papers based on publications, what is going on in other countries, advanced level of research, what is going on. So we from there a lot. But coming to a program like this, you have first-hand experience with an expert, and we are going to have a conference here from 4th to 6th. So there are a lot of experts coming nationally, internationally. So there also you can interact with people. So first-hand interaction, that is, you cannot replace that one. You will read a paper and everything, that is fine. And you may, even when you submit a paper, you get comments from experts. But when you are actually giving a seminar, the kind of questions you get on the spot, because you have to answer, you have to think immediately, right? And you also get a lot of ideas. Because someone is asking a question, it makes you think in a new direction, right? And now we have experts who will talk about their research background and all. You can also ask questions related to your research problem. So you will get some input that is not possible to get you from other places. So this is a really great opportunity for you to learn here, and more important is go back and implement at your own institution in your research. And also you can spread the word with other researchers in your institution who want to work on plant metabolomics. So give them the knowledge that you have gained. So you need to distribute it, right? Spread the knowledge. That is the best thing that we can do. And maybe you will get up with new collaborations. And we have an expert like Professor Zhao. We can have collaboration with him, our university, if he can find something that is common. And nowadays there are a lot of international grants also that originate from India and also outside. So these are the opportunities we can try to build up. So I hope this workshop of say 1 week will really give you good insights into not only theory, but there are also some practical sessions, right, going on here. And you can also interact with other faculty, like plant science faculty are there, others who are working maybe in biochemistry, pharmacology, who are working in related areas, or others. You can also interact with them. So Dr. Vinay can probably help you if you want to interact. And Dr. Vinay, you need to also get pharmacology faculty to interact with Professor Zhao, because Professor Sanjeev was saying his background is pharmacology, right? So we have a pharmacology department here who are doing really well, and pharmaceutical sciences also, who work with plants and all that. So that would be good to interact with them also in some session wherever possible. So I hope you all will get a lot of good opportunity or information from this particular GYAN program and benefit from this. Okay, thank you.
So thank you, Professor Ramakrishna sir, for explaining the different disciplines, education policies of our university with respect to the academics activity. Yes, there is a chance to interact with him also with multiple dimensions. And yes, we officially are starting this live is GYAN is going on, but yes, after 10 minutes Professor Jeng is starting his presentation. Before that, I would like to vote of the thanks by Professor Felix Bust for this inauguration session at least. Professor Felix.
Well, hello everyone. Professor Zhao, welcome to our university and this GYAN program. And without further ado, I've been asked to give you the vote of thanks. So first of all, I thank Professor Zhao for coming all the way from Hunan. Today morning I had a small interaction with Professor Hunan, and I came to know that he's from Professor Zhao from Hunan. Hunan is a place somewhere right in the center of China. And it's so beautiful. You can just check out some of the pictures. Avatar, the movie, was shot, some scenes from that place. And I also came to know that Professor Zhao, sorry, get confused. So, Zhao worked in Kyushu University in Japan, where he did his JSPS program. And yes, I was also somewhere close to the Kyushu in Shikoku. I'm looking forward for further interaction, sir. Because this topic is really important. As you know, the plant metabolites are super important for commercial point of view, agriculture point or medical point, isn't it? Most often we think that the modern medicine is all synthetic, isn't it? Not really. You see that any medicine that you buy from, the pharmacy, for example, almost 90% have got plant origin. And most of them are coming from secondary metabolites. And if you know how to make it or how to boost the production of the secondary metabolites by genetic engineering means that Professor Zhao is an expert in it. You can drastically improve the quality of the food because I think he's more into the agriculture point, the nutritional enhancement of the food, and also for pharmaceutical and nutraceutical improvement. So this topic is really important, especially with regard to the IPR and for commercialization and plant biotechnology. This is one of the crucial topic in plant biotechnology. So yeah, and also in this university, as I hear, well, at least in botany department, we never had a foreign faculty coming to give a training. So yeah, congratulations to Dr. Vinay and also to Dr. Mukesh for pushing all of us to submit a lot of applications, and glad to know that 3 of the 12 applications was successful. So, well, ultimately, this program is going to be really useful for those participants who are right here, who is coming from all different places in India. As I hear, 22 participants are outside the university. And yeah, so welcome everyone to this university. Ours is a new university, kind of new, 15 years now. Yeah. And you're welcome to visit in case you're free. If you really want to see around the university, you're welcome to come to our labs. We'll be more than happy to take you around, right? And see our CIL. We have a great facility there. And yes. So thank you so much, sir, for coming, Professor Zhao. And also all the 22 external participants, I think, we have to formally invite you to this program. And so I really thankful to all of you to come to this program. I hope you will be coming for the next GAN, incarnation. So I hope we are going to have more GAN programs. 2 more is in pipeline. And also just before you go, please plan to attend our conference, right? This program is till 3rd. Now 4th onwards, 4, 5, 6, we have an international conference coming up here. So if you can extend your return journey by just 3 more days. So you'll be part of our international conference where so many international speakers are coming here for speaking. Okay, so you may please consider it. And I'm also thankful to everyone, especially the patron of this program, Professor Raghavendra P. Tiwari sir, our Vice Chancellor, and also the whole team behind this program. And last but not least, Ministry of Education for giving us this grant. The GAN is a very prestigious grant. So congratulations everyone, and thank you for coming, sir. And looking forward to interact with all of you. Jai Hind, Jai Bharat.
So thank you, Professor Alex. Professor Alex is an international faculty in our departments already. Yes, I would like to introduce another faculty of my department who is very promising in the potential with respect to the agriculture genomics and transcriptomics. So I would like to invite Dr. Pankaj sir to give a few words about our departments as well as from research in which our department is working.
Good morning. Welcome, Professor Chow. I'm Pankaj Bhardwaj. Okay, so I know her basically. She is one of my junior from Chandigarh. Welcome. I welcome all of you to our campus, to our department, Central University of Punjab. Primarily, our department focuses on algology, and my lab is primarily focusing on molecular genetics. I work on evolution and adaptation. That is one of the areas which I am working. Then Professor Sanjeev, he primarily works on stress physiology. As far as the metabolomics is concerned, I think all of us, all of 7 faculty members, somehow we are connected to metabolomics one or the other way, and that's why in our university we have GCMS, MS, and another one which is another instrument which is LCMS, MS is under the pipeline. All of you, whoever wanted to, can have all the facility, access to all the facilities. You can primarily, look around. You can go to CIL, and whosoever is working around, you can have the discussion with all the possible faculty members here in the department, not in botany only, but other faculty members, say like if you wanted some of the faculty members who are working in molecular genetics, molecular medicines, then pharmacology, pharmacognosy. There are many of us who are primarily working on this particular situation. You can have discussion around the subject. I can elaborate around the work which is primarily being done by my laboratory around metabolomics. So one of the work which we primarily took up was an Himalayan plant which is called as an rhododendron. We were primarily very much interested in looking into the metabolomic landscapes and transcriptomic landscapes which are changing with the development. So those are the particular papers which we have worked a lot on metabolomics. Though I am not a trained metabolomist, frankly speaking, but we have gained the knowledge in due course of our academic journey and the research journey. If you have any question, I will be here for next 5 days. I'm really looking forward for the interaction with the Professor Jao, so we can have a future collaborations. We can really work, we can come to a very common ground where we can have fantastic collaboration, not to I mean, the work which we are doing, but the work or a common ground which we can have in the near future, say like for 10 years or 20 years coming. So that's what we are looking ahead as a department, as a faculty from the botany department and specifically who works in the genomics. With these words, I welcome all of you to our department, to our university, and I'm really looking forward for interactions with you guys. Thank you so much and welcome you all.
Thank you, Dr. Pankaj. Now we are announcing the first lectures that is going within the 5 minutes. After this first lecture, we will have a tea break, but I would like to invite the Professor Jain to start the first lecture of the Gyan. Yeah, please, sir. Professor Jain. So, my point. I would like to, yes, Dr. Mukesh told me that we have to submit a 2 proposal. So I submitted the 2 proposals 1.5 years before, and one with the Russia and second with the China. So I missed the Israel and others faculties I know very well. So my 2 projects were, both are recommended, but one was strongly recommended by very top-notch scientists of our country, even the IMS directors and many other persons. Second was only just recommended. So finally I got the news that we are only giving the courses which is strongly recommended. So when I saw the peer reviews comments on this plant metabolomics and the technology, then it was strongly recommended by 3 members, different anonymous members. So because I know this technology is really very useful with respect to animal sciences as well as the plant sciences, and as a part of Department of Botany, we propose these courses actually. And yes, in the near future, it's the era of small DNA transcriptomics that is now come up very advanced stage. So next question is come up that how this gene influence the phenotypic appearance of any organism. So if you go and find out what is the channel between the final phenotypic appearance and the expression and regulation of those genes, so you will identify that there is some metabolites which directly or indirectly intermediates of the different biosynthetic pathways we finally come up by the different enzyme catalyzed reactions which are encoded by the different genes. So final end thing which influence the phenotypic appearance of any organism, adaptability against the different situations, so it is the metabolites. And yes, I would like to request, I will take many lectures and you will understand many things in coming days. And we also arrange a few surprise for all of you and visit with the CIL also. We have GCMS, LCMS, HPTLC, and another important points that is we have the NMR also. What type of the data you are getting, okay, how you analyze them, what is the best way to get some meaningful information from those graphs. So we will discuss those things with all of you, and now because I'm getting very good response right now, so I'm thinking to conduct a workshop in the March and April at the national level also in the same or the similar topics. So now I would like to request the Professor Jeng to start the first lecture of the Gyan, And please mind it, it is the live all over the India on the GAN channel right now. And yes, Professor Jain, you start. And the lecture is around one duration, but it depends on the potential of the Professor Jain. But you have to take from here because there is a mandatory situation actually. We can't move. We have to stand here and just explain the things. So this way is okay, but that thing is not possible according to the GAN mandatory instruction. So that is a disadvantage for me also.
It's really my great honor to be here to, just talk about the research topic in my research area. And really appreciate Professor Ravindra Kumar and also Vice Chancellor of the Central University of Punjab give me a very strong invitation letter to support the e-visa application. Actually, when I apply for e-visa online, there are several some troubles. Actually, I applied twice. So the Vice Chancellor, the Professor Tamari, the strong invitation letter is very, very effective. Finally, I got the e-visa, and he enabled me to come here. Otherwise, and also Dr. He knows those troubles. But eventually, we succeed. I'm here, so it's really kind of an honor to be here to communicate that. I'm really present our university because from Vice Chancellor, the invitation letter and our president, university president also look at the invitation letter and see, okay, if we can establish the relationship, very close academic or research relationship with Central University of Punjab, that's going to be great. In the future, we can exchange some new idea also. So, about the metabolomics, actually in past like 20 or 20 years, I mainly work on the metabolite from biochemical side, the isolation and the characterization of chemical structure, and then I transfer to molecular biology, but still depends on the metabolite profiling. But with the tool of molecular biology approaches, so I can understand the below the metabolites change. Plant phenotypes really depend on the metabolites. Just like very common, we know the diabetes, or we know the the disease actually caused by uric acid accumulation, overaccumulation, the kind of metabolites, right? Overaccumulation causes pain of a junction area, our knees, so those actually already started very well in medicine to understand the cause reason of this disease. This is very earlier the metabolomics start. So anyway, that's why I initially studied biochemical and phytochemical. Then I developed the molecular biology. Now I actually work on both. Use the molecular biology tools to understand the plant phenotype and the phenotype related to the metabolite, and the metabolite related to the crop nutritional quality, and also the mechanism underlying the various like vegetable or horticulture plant, the nutritional quality, like the different taste, right? Like the sour or sweet or bitter or any kind of really just like Vice Chancellor, our deans, our professors can understand the recent years, metabolite really heat up because those metabolites related to not only the medicinal plant, but very earlier, many human medicine actually developed from plant natural products. But also now, enhance our human health, our nutrition. We found not only related to taste, also related to the flavor, what we can smell, those volatile or non-volatile metabolites. So overall, I just give the whole concept of this our study, like systems biology, our even in genome level, systems genomics. That's the frontier of those study. Try to look at the crops, look at a certain plant from a whole view to see the plant evolution or domestication, and then develops the current phenotype. How the humans select those, like rice, our soybean, we select the rice. Actually, it is very different from the current rice cultivars, very different from the wild rice or some intermediate land races. So but in those systems biology or systems genomics, we first see the phenotype. We first see phenotype. Doesn't matter horticulture plant, we taste sweet, or we smell very good, or various. But we can see many different the phenotypes. Then below that is the metabolite, lipidomics or ionomics. There's some plant probably we don't know the ionomics. But in recent years also the ionomics become also important. They are developed by CTMS. Later I will describe all those different omics, metabolite omics, how to use the different tools to profile, then to understand related down the under part, like metabolome. Of course, those just a name to collect, to name all those omics, metabolomic, lipidomics, and also carbohydrate, also proteomics, those larger molecules. But for metabolomics, we only usually focus on the 2,000 kilodalton, those small molecules. And metabolomics, of course, are a subject of science. The proteomics and transcriptomics and genomics. So the different level. But most often, we use the transcriptomics. And recently, people study the proteomics. Because in medicine plant, in medicine, the human medicine, more people study the proteomics or protein levels to understand disease development, and also there's some cancer also. To find the target protein, then find small molecule to interfere with the target protein. So to establish the medical model. So, but in plant, we still feel very difficult to study plant metabolism from protein levels. We only study some from enzyme activity, but those still you can see very little effect, very little work, because work on protein much more difficult than working on RNA or DNA. Because those already tools are very sophisticated. You can profile, use different kits to study, but protein is still a kind of and also people like University of I think the University of California, Riverside, the academy member, Steve, he found the transcriptome in plant, transcriptome and proteome, there are huge difference. The matching degree is less than 50%. That means a lot of alternative splicing or some different proteins. You don't know where it come from. You cannot match with the transcriptome, message RNA or any the transcripts. So those still are mystery between the. But however, from a transcriptome, usually from can almost cannot say perfectly connected with the metabolite. Usually we saw this gene expression, and we can see the related the enzyme, encoding the enzyme, the products accumulation. Yeah, they can find a good correlation. When it comes to the metabolome, I guess those knowledge you can see from the website. The very earlier, they found how they initially started this metabolite. Just like when we go to the hospital, we want to find what's the disease caused from, or what's the reason. And usually, in China at least, the doctor will make some respiration, analyze the urine, analyze the blood, and do other different tests. Many of those tests actually, some are biological, like counts the blood cell density or number, but some of you try to analyze your urine content, and what's this almost become the basic prescription text. So actually, initial the work is just from patient with the pain in the junction, and the doctors found doing the metabolite profiling found that the uric acid accumulates very high level, and they find this level. This is just because of the uric, and the defective in further catabolic uric acid to discard, so overaccumulated in blood. So basically, the concept is that, I saw that, but anyway, there are some the Chinese character, the small molecule, the molecular weight less than 1,000. The concept of metabolomics is the quantitative the measurement of the low molecular weight metabolite in one organism or one cell at a specific time on a specific situation. That's a temporal spatial. We call temporal spatial because every moment the metabolite is changing. From circadian rhythm, we have day, we have night, so those change. So those are the rule of change. They have a lot of meaning. Yeah, that's why we, in medicinal or in plant, we study those metabolomics. So from another point of view, from a subject, we see the metabolomics is a quantitative description of internal all the metabolites in a whole living cell or tissue. Try to emphasize in the inner those change and also outer change in environment change, how the environment change affect the inside metabolite. Our inside metabolite, our inside the gene, our the expression of protein of some the functional defect can affect our metabolite, then reflected in our phenotype. Or in human, we have some disease, uncomfortable, or different kind of. That's from a concept. This is the development history of metabolomics. This the timeline from 1982 to define this urine profiling, the publication, and connect the urine profiling, and find the high uric acid accumulation, the very strongly correlated to junction, the pain. Yeah, finally they established, and also use the NMR. I guess Dr. Professor Vinu will talk about, so I will mainly talk about the mass spectrometry. So until now, the already very, very sophisticated approaches from non-destructive, like NMR profiling metabolite, and to some very fine, very sensitive profiling of the metabolite from a non-target. One time can profile the several thousands of the metabolite in LC-MS or LC-Q12 to our targeted. We only try to target some part. You use the targeted metabolite profiling, specific in the extraction part and also in pre-treatment of the sample. Finally, to apply it on the LC-MS or GC-MS. So you only focus on those targeted, a group of metabolite. This you can really zoom in to see the more detail of this kind of the metabolite, the targeted and non-targeted. So already very far, already exists too much from those earlier publication or earlier development. So I will just quickly go the sample preparation. Actually, later on, we can specifically describe the extraction or the sample preparation, or some sample need pre-treatment, or pass the pre-dissolved or use the pre-column to treat, to remove some other metabolite or other the solvents or some to interfere, to minimize the interference of those substance or targeted the metabolite. So like first we extraction, then probably we need a pre-treatment. Sometimes you need a desalt, sometimes you need to pass a pre-column. And then we do the separation. The separation have different ways, like HPLC or UPLC, or use the GC, have very long, the column, for the different the volatile or non-volatile compound. Then for detection and identification, the later we will in each specific the machine we will talk about. Then to the data analysis, sometimes we will do visualization. We do the finger comparison, or also recently there's some new mass spectrometry development to specialize, visualize different metabolites, use like the heat map, those technique, and then do the modeling, do simulation. There are very different type of. So the correspondingly, the bottom shows the different extraction. There's solid phase micro extraction, and also the solid phase extraction, or some other affinity chromatography. And sometimes for gas spectrometry, and also for spectrometry. And mass spectrometry, and NMR, or some mystery. Those for depend on the electrochemical. And analyze use the bioinformatics or chemical information. So to understand why we study the metabolomics, just because our whole life is actually a kind of a movement of chemistry, right? Our body, all the world is composed of different the chemicals. So actually we study metabolite. It means that we understand the life is based on the chemistry. And certain chemicals form the life activity. Yeah, that's really. And also, below this, in human, our life activity is like a controlled, not like a regular mechanical composition. They arranged in very different orders to form a different life style or life forms. Therefore, like genome, the encoder encoding the genetic information, and genetic information then use the transcriptome to transfer into the transcripts. The transcripts then translate into the proteins. Then protein to conduct the enzyme activity or conduct the structure of the cell. Inside the cell, there are some actin or tubulin fibers to build the structure inside. To the cell actually there are uric cell. There are plasma membrane, and inside there are nuclear and also other different organisms. So those need their support. The support is like fibers, the different like a sinker fiber, like a tubulin. And on the tubulin, there are some actins to move those organelles or some proteins and of course, inside the cell, people sometimes you see the video. They said inside is like a gel, like a gel. Yeah, very sticky. But in certain part, is not evenly sticky. The certain part is very active. Now there's some. The biologists study the inside the cell, the structure, or even the nuclear structure, or even the genome structure, the 3D structure, how the genome, for example, metabolite, for example, flavonoid. All people know flavonoid biosynthesis is very quick, but how those quick things can happen like in 8 hours. In 8 hours, when we stimulate or we light or use some pathogen stimulated plant, we will see the flavonoid, certain flavonoid de novo biosynthesis happen. Some people say, oh, there's some pre-existing precursor there. But actually, when you check the messenger RNA, you will see many of them is de novo biosynthesis from beginning. In a very short time, how the accomplishment, the quick biosynthesis of this metabolite through this more than 10 different enzymes compared. So there's some the biological secrets, the coding information, and from this epigenetics, the metabolite, and the phenotype give the defense. The plant start to defense. Some very sensitive, but some is resistant. Some plant is resistant, some is sensitive to give different phenotype. Of course, we don't need to talk about the essential necessities. Very important. So about the application of metabolite, you almost can see it everywhere right now. So you see a tremendous application in different, not only the medicinal plant, cancer, kidney, urological, inflammation, or immunology, or respiratory, like COVID-19, the different people actually in 2000, from 2020 to 2000, now people still use their some sample, old sample, to study how the virus, the COVID-19 virus infection change the organ, the lung, the structure, or change the metabolite. Actually, all of those, the metabolite in this case will give you a different angle to look at the virus have destructive effect on the lung, the function. So those, I believe, will continue from metabolic gives our insight, new insight into the COVID-19 or some even in future unknown, the deadly virus will how impact on the human function, and also others. From a plant the point of view, I will mainly talk about the immune plant aspect. Clinical metabolite disease diagnosis, treatment, and also depend on the metabolite analysis result. Doctor can quickly diagnose what's the problem could be, and then try to find the curing strategy. And medicine, of course, absorption and also catabolic dynamics. Every new medicine come out, we have to use the volunteer to take those, then quickly detect our plasma. And in what time the plasma, this medicine will reach the maximum level, and then degraded, catalyzed, degraded by our then also there's some toxicity. When we studied even the traditional medicine, like the traditional Chinese medicine recently, later I will talk about the some medicine initially we don't know. But long term, the clinical examination then finally they know there's some traditional Chinese medicine even used like 1,000 years, but eventually found there are some carcinogenic the organic compound. This initially thought probably is like a bioactive compound, but actually is a carcinogenic effect and cause the failure of kidney and the liver. So I will later we will talk about how the metabolite profiling is important for the pharmacognosy. And the nutrition, we also, when people study nutrition, also use the metabolite everywhere. And plants and crops. We use nutrition of metabolic pathways, characterization of function, and function of metabolite enzymes, regulatory factor. That's why many people try to use the regulatory factor characterization to one step can control the whole pathway. Because usually, just the minutes I said, the many stimulus on the flavonoid biosynthesis, the effect is very quick. So how this happened? Some of the reasons. There are many reasons, but some of the reasons is about the transcription regulation. The transcriptional regulation can very quickly control the whole set of the gene quickly to finish the de novo biosynthesis, then translation into the enzyme, and quickly those enzymes form a metabolome, an enzyme interaction, and form a metabolome from the starting substrate, and quickly go like a metabolome channel and gets the final product. So those will be much more efficient. But many scientists study regulatory function just because they believe the metabolite, the channel, actually is working. some of them we don't know. Like recent last 10 years, like Joint Center, the Anna Osborne, Professor Anna Osborne found tritropinoid and saponin. One of the tritropinoid is saponin because the saponin actually in Arabidopsis and also in oats, the genome, they assembled like the metabolome. Those genes, the correlated genes, or key genes, they cluster together. Usually those kind of situation only found in fungi or in other prokaryotic the bacteria. The some part like metabolite, like sugar, metabolite sugar or glycolysis enzyme in bacteria. So they form the metabolome. They form a regulome. Regulome is just one transcription factor that control all of those genes. Of course, in bacteria, the gene is overlapping, right? They're overlapping. They don't have an intron. They really, but they synchronize the genome. That's why their genome is small. However, they can encode much more enzyme than we expected. So they can very economically use those limited genome size to encode the much more enzyme to finish the life cycle. So that's why the regulatory transcription factor is very important, and also underlying regulatory mechanism. Those can facilitate later on our improvement of food yield. Yeah, that's a very significant study in the American or some, for example, in USA, when I once studied proacyanidin. The proacyanidin is one of flavonoids. It's similar to the proacyanidin, but it's a different branch. From flavanol, then all the way go down, there's some two branches. One is proacyanidin, one is acetylcysteine. These are two very common, most widely distributed flavonoids in the plant kingdoms. Those transcription factor, we found that one transcription factor can control proacyanidin production. This production actually the benefit in the alfalfa. Alfalfa is a high quality forage, right? But however, their proteins so quality very high, very easily digested and generates the methane. So that's why they kill our sheep. When they sheep, they have two stomachs, right? The first stomach, the second stomach. The first stomach, when they take up the alfalfa, then the protein quickly digested, then generates much more methane. If they cannot quickly transfer or digest too much, they take too much alfalfa leaves, then will generate too much methane, then bloat. The bloat then the animal causes death. The cause pasture, the lethal pasture, the bloating. So the proacyanidin actually can prevent the protein degradation, protein in alfalfa leaves degradation. If we mix the alfalfa with some pine's bark, the powder, or some part mixed together, they can very effectively prevent the lethal pasture bloating. So reduce the agricultural loss. However, if we can theoretically manipulate the proacyanidin production in the plant, and that's alfalfa accumulate some part of some level of pyrocyanidium. So those, when we do the grazing, just grazing, let the animal to eat by themselves. So we don't need to worry about the lethal pasture bloating. So that way, we can improve not only yield and also nutrition quality and metabolic engineering of useful metabolite, either for medicine or for agriculture purpose. So for human medicine, this I guess we already mentioned. We can identify or quantify all the metabolite in a biological sample. That's now actually in China, or at least we know there some professor joined a company because one professor does metabolite very good, and of course they're also managing the university's facilities, several different kind of Q-top or extra, just thermal Fisher, the very high quality of metabolite profiling or some chemical identification. Use this machine, you can help the patient to quickly profile the biological sample from patients that finds is there any difference, and also understand how those metabolites change in response to the various factors, and provide insights into the physiological state of the cell, tissue, or organism. If we know the normal, regular should be like every parameters of a substance or metabolite should be like this. But if you have lower or higher, that's how we. I guess you hear also every year your faculty member will do the examination test, right? There's a lot of items to be examined. That's how they try to find it. But some of them just try to detect the antibodies or the. But anyway, those are very significant application in the plant pharmacology, biosynthesis, medicine plant also. The transport accumulation pattern of in different tissues and the plant pathogenic or plant pathogenesis. And we know the secondary metabolite is very important for plant to defend against the various abiotic or biotic stresses. And some of metabolite like caffeine in tea plant actually and immune system, they can defend against pathogenic microbial or against the insects. So there are many different type of metabolites, secondary metabolites, mainly for the function of against those abiotic stress. The food safety also have a significant application of metabolomics, like agrochemical analysis, the pesticide, herbicide, fungicide, or heavy metal. We also can detect when we try to export some food or some stuff to other countries. Usually, other countries won't detect it, like we in China. We export a lot of tea, green tea. However, they want to test whether your agrochemical, the overaccumulator, even we always pursue the organic, our green cultivation, the green products, organic products, but still there are some steps. If the contamination happens, then the products just cannot be accepted, have to be returned to it. So this is very huge loss. So the target of metabolomics, we mainly have a following part, like a carbohydrate. That's very common. We try to understand like plant yield. The plant yield is very important target. We try to increase the yield. However, how we understand the yield, the limit. So just from the carbohydrate photosynthesis, how many the carbohydrate you can produce by photosynthesis. However, how many energy our C2, C3, those metabolites, those different type of photosynthesis in like corn or in the rice, they're very different. So the photosynthesis efficiency is different. However, through the profiling of the carbohydrate, simple sugar or some sugar modification, phosphate sugars, you can actually understand which part is the limited part, metabolite. This carbohydrate profiling not only understand the yield, but also understand the recent year the biofuel development, to try to use the cell wall to hydrolyze or understand how to reuse the cell wall, reuse the stem or waste the biomass. But we know it's very difficult to reuse because there's the central recipient, some lignin inside. They cannot be hydrolyzed by the fungi. Very difficult to hydrolyze. Lignin is very difficult. But if we reduce the lignin, what's happened? That's why like in past 10 or 20 years, the biofuel, the hot, is just because we're concerned if we petrol oil or the fossil oil will contaminate our environment, global heat emission, or whatever those concern, we have to turn to the biofuel. So those carbohydrate, the profiling also to understand the cell wall structure, how cell wall structure change. If we manipulate the biosynthesis gene, the change of the cell wall structure can affect the later on the feasibility of digest or hydrolyze use other fungi. Yeah, you need these great achievements already being made to understand the cell wall structure. This is like a carbohydrate profiling. And the many different simple sugar, but also currently people can use the different profile to do the larger sugar, larger polymer, carbohydrate or saccharide compound. And lipid is also recent years already gave very significant achievement. So now we understand lipid already is much more advanced. We know the lipid not only affect the food quality. We know that some rice you taste very good, and some rice you taste not very good. Those are mainly related to the lipid. Now we understand. Yeah, because lipid, since it's a metabolite, can release some carbon 6, carbon 9, or other carbon 18. Those fatty acid metabolites makes the rice smell very different. Yeah, like Thai, the rice, right? Yeah, it's very. So the lipid actually, we later on probably we can talk, and because I'm in USA study a lot of lipid. If you guys some of the study lipid or interested in the lipid, not only for the oil, the vegetable oil biosynthesis or improve the production, or just for the other purposes. Like in USA, there are another way to do the lipid biosynthesis. You try to produce the hydrogen, hydroxy, the long fatty acid with the hydroxy group. So those oil, from a chemical crop can use it for lubricant, right? Can use it for the high quality of lubricant applied in flight or others. So those different application, and also for the nutrition purpose, people try to produce the long chain, the fatty acid, omega-3 fatty acid, like DHA, EPA. We know DHA, EPA, right? Like the fish oil, they have this kind of like a carbon 13, carbon 20, carbon 22, carbon 24. Yeah, those long chain fatty acid. I know in England, they try to produce this long chain fatty acid in rapeseeds. Yeah, here grow rapeseed, right? Yeah, grow rapeseed. They try to produce these rapeseeds inside can grow the long alpha fatty acid, so everybody can get the benefits from eating more and beneficial for brain, right? Yeah, that's all. How I think later on we will, and also fatty acid. When we study the protein or study the high fatty acid, the green, like we know some fatty acid is essential. We cannot synthesize, our human body cannot synthesize. We have to take from plant food or from meat. So those fatty acids become essential fatty acid. So in rice, in corn, the lysine is very low. But in alfalfa, of those forage, try to feed like a sheep or cow or other animals, we try to use their wool, use the fur. Those actually we expect the higher cysteine. High cysteine level in alfalfa is very highly anticipated because those high cysteine can improve the wool quality, the sheep, the wool quality, and also others. So that's why some, like for the lysine, high lysine corn, and I know one American Academy member worked on corn for many years, just to try to improve the lysine content in corn and also cysteine of those sulfur-containing amino acids in forages to improve the food quality. Organic acid is also very common. We do organic acid not only is central for the protein or for sugar or for other biosynthesis, because we know this. If you study the oil biosynthesis, for example, rapeseed, soybean, or other this oil biosynthesis very, the fatty, those organic acid very important. There's some transferrin factor directly regulates the fatty acid generation from glycolysis, then quickly directed to the fatty acid biosynthesis, because this is like central. I think later on we can have a whole view of the metabolite profiling, how these are meaningful in animal, in metabolomics, beneficiary to the plant science research. And then also the vitamins, we know have different the purpose. Some vegetable we try to increase the vitamin C or increase the vitamin E those efforts already have products in market, like pre-vitamin A, the golden rice, and the hormone is very important. We know it regulates the physiological process. Every hormone, we try to understand. Some hormones stimulate the flavonoid, some hormone, like jasmonate, usually stimulates secondary metabolite. So in the caseosus rosas, we try to produce Christine, perchristine, and anti-cancer compound. We usually use the jasmonate to stimulate those indoor alkaloid the production, and also supporting very effective. So the hormone also connected to secondary metabolite. We usually do hormone profiling, use targeted metabolite method, use some internal standard. Also, the secondary metabolite were not too much. Then iron. Iron recently, people start to pay special attention to the iron, not only cation, like heavy metals in our food, the food, our environment, in soil. Like recently, we have nationwide scanning of all the area soil, because in the past 30 years, those very quick development of industrialization of many the mining. The mine is developed to produce some color metals, the different metals widely applied in the industry. In China, particularly the southern part of China, like Hunan province, we are living in. There's some mining contamination caused, and also the China, when you burn soil, it burns some soil to solidify, to produce the China, right? To produce the different kind of tools. I think they call China or ceramics. The ceramics, right? Yeah, ceramics is more common. But China also is another kind of name. They're everywhere burning those. But the burning those can release a lot of fluoride into the air. The fluoride contamination, actually recent years globally increased a lot. So we study tea plant. I know pretty much because a lot of tea, tea plant is a very hyper fluoride accumulator, hyper aluminum accumulator, because the tea plant usually grow in the Aztec region. The soil Aztec pH like below 5, pH 4 points, some 5 points is better. But in those soils, they quickly absorb the aluminum from the tea plant very tolerant to the aluminum. You cannot imagine, like in Arabidopsis, 50 micromolar, the aluminum can cause the root growth inhibition, severely inhibited. And also in wheat, the same. They also inhibit the wheat growth. However, in tea plant, they can stand like 2,000 millimolar. 2,000 micromolar. No, like 2,000 micromolar. It's like 100-fold the tolerance. So the mechanism is they quickly take up the aluminum, accumulates in the old leaves. In tea plant old leaves, they accumulate. Also fluoride too. Fluoride accumulates quickly in tea plant in old leaves. But also in young leaves, they also have some minor fluoride. If you consider the recent global temperature increase, and we air the fluoride increase, and now we don't need any fluoride in our toothpaste. You know, the company sells the toothpaste before advertise, okay, the fluoride is the beneficiary. Yeah, before the beneficiary, even UN have an issue called every day we need, like adults, we need to take a 4 microgram per day to meet the fluoride requirement in our body is essential. But however, now it exceeds much more in this level. From food or from other, already. So you know right now, you don't see any toothpaste. See, okay, we have fluoride. No, you don't need to see. You don't already. We already have much more enough. So that's all those ion, cation. Now getting to the people's, the patient. We need to consider the health. Analytical tools of metabolites. This, which is first general described. Later on, we will focus on some most commonly used, like infrared IR spectrometry. That's now we still use for structure determination of chemicals, and also nuclear magic resonance, the NMR. Probably Dr. Winner will talk more. And thin layer chromatography, now in our lab still sometimes using to isolate specific compound to very useful, still very useful in many chemical lab. And HPLC, this is very more common because we basically already use this as a regular tools to separation and mainly test separation and quickly detection. Use the standard compound to and also those if coupled with different detector like UV detector or the photodiagonal array detector PDA, so you can scanning from 200 to 800 nanometer the solute to pass through, you can scanner to find those, particularly when we start a metabolite, those colorful carotenoid, you can use this PDA. Basically here can identify the different flavonols. Flavonols have maximum absorption at 350 around nanometer, and acetylene have 530 around, and also other like flavanol, other flavanol catechins or isoflavone, they have 207 or 200, even 210. Yeah, those nanometers. You can use the PDA can quickly primarily quickly judge what compound it could be. Yeah, those. Actually, this later on we will talk more about this because this is very common. And even UPLC, the ultra high, just gets very faster. The previous HPLC, one sample, one program isolation is 20 or 40 minutes. But now use the UPLC, you just need like 5 minutes or 20 minutes. The correspondingly short and also resolution is high. You get the very sharp peaks and the separation of the molecule. Even though we still know that the HPLC, 1 million HPLC doesn't really separate all because they still have some compound mixed together. But anyway, for major, for those very reproducible, for example, we know now HPLC is kind of reproducible. So even some still say those not very. as long as you detect those high level amounts of the metabolite, like when you analyze the catechins in tea leaves, you always get those EGCG very high, and then EGC less higher, then ECG is much more than EC lower. Yeah, you always get for the cultivar. Doesn't matter from India or from Kenya or from China or from Japan. Yeah, those cultivars are very similar because, of course, there are variation because the people, the domestication or in different region, they have a different favorite. For example, we favorite less catechins, but we want more flavor, even more smell, the flavor. So we want to smell the very bad, good, or different flower, or different kind of a fruit smell, but we don't want to taste too much astringency or the bitter. However, in Japan, they expect to taste astringency and bitter because they think that is more healthy, the better. So that's really depend on the. So just because the different favorite, the plant or different cultivars clearly rely on people's selection. So now we still on the track of domestication, tea domestication. We can see clearly because I'm studying tea. Later on, if you guys are interested, we can see. For example, we collect some tea from Assam area in India and also from Yunnan, Guizhou, and different area, different wild tea relatives, and we can see the change of those metabolite profile. It really depends on our utilization, how we utilize. Also soybean. The wild soybean is black seed, but after also white rice, wild rice is black, right? The seed coat is black. Soybean also black, but after 1,000 years, people utilization 18 and felt that black not good, taste not good. And suddenly the genes mutation, the farmer found, okay, oh, my soybean or my rice is white. Oh, that's beautiful. Then they collect, then they grow up. Then they collect the whole, of course, they might have segregation. The next generation, they found out, okay, some will become black again. But most of them still the white or no color. Then you just collect, finally found, this is the domestic selection also depend on because as sanguine decrease, definitely oil increase. Oil content increase. That's when we study the oil biosynthesis, we know the seeds color affect oil seeds, the oil content. So when you see the oil increase, you taste good. So you both good looking and taste good. Why not choose this one? That's why we collect, we get this selected, this trait selected. Finally, we know this trait selection, the mechanism is there are TT8 gene mutation. And also there are CHS, the gene mutation. But the different region, you always found there are different mutation. Finally, people collect all those, the different colors, soybean, then do the QTL mapping. Found there are at least the 8 or 6 genes underlying the seeds, those pyrrolizine or ascending loads. So those just according to the ascending profile, then quickly get find the the color caused by cellulose, the segregation, co-segregated with one genome loci, then the mapping to the original mapping to a fragment, a certain area, which centimeter or to which marker. And finally, after this genome sequence, or even some great scientists mapping to the single gene, and they found the TT8 or CHS, the inverted repeat, and cause the generation of small RNA mutated to other CHS homologs against the whole seeds, CHS, the functional loss. Then the seeds become the colorless. So all of those, I think, are very interesting. Just use the metabolite to provide you can get you can see our human civilization and the plant, all of the crops, domestication, the process from the phenotype, metabolite, all to the current, the cultivar established. We know the back behind those, and also like strawberry, probably local strawberry. In USA, strawberry large and firm, have a long shelf life, can store it for long. This is just for commercial value. However, it's not for the nutrition or not for the taste value. The taste is not good. It's much worse than some local strawberry. Local strawberry, when they get matured, the taste is very good, juice, and smell, also have smell. So I see the article in Nature said why the strawberry, because strawberry is a very high valuable, worldwide the fruit, right? The very high value. People try to understand why during the strawberry domestication cultivation we lost the smell, lost the flavor, very good flavor, very good smell. So what's the loss behind it? They come back to the wild strawberry and compare to then we find the human selection for one purpose. Meantime, it's low starter. Yeah, that's how very interesting. Then I will just do quickly. The capillary electrophoresis, the UV, ultraviolet absorbance detector, all of the coupled. The coupled means use capillary electrophoresis, the high efficiency separation, then coupled with another feasible detection system, the separation system, and coupled to detection system. But the most efficient is still the capillary electrophoresis coupled to the mass spectrometry. And just chromatography mass spectrometer. This also very common. I think later we will talk more about GC and LCMS because those are much more commonly used. Below, Fourier transform ion cytron mass spectra, this is not the common use. Some machine put it in the lab. The whole years nobody touched that, just because one way is the technique required, special technique required. Other way is that people try to get used to the other familiar machine. They don't touch those the hard manipulating machine. The AAS, still now we analyze, we use a lot to analyze the metal ion, the atomic spectrometry, and also ICP-MS, the inductive coupled organ plasma mass spectrometer. Yeah, those are for different purposes. For the comparison of metabolite tools, NMR or NMR is the kind of non-destructive. You don't need to destroy the sample. You just put the sample into powder. You can directly test those. For example, what I used was use NMR to analyze. So the one compound coupled with aluminum in tea plants. Yeah, we know the tea plants accumulate a lot of aluminum. However, what's the form of the aluminum exists in tea plants? They usually combined with organic acid, citric acid or the malic acid together with aluminum, like the three charge, right? So three covalent charged, so they can combine to form a complex. The complex people know to is one way of detoxification, the plant detoxification. But I use NMR to detect really in tea plant what's the organic acid, the major organic acid to chelating the aluminum. I finally found the citric acid in different organs, different. For example, in root, the malic acid. So the chelating the aluminum. So how to do that? I just use the whole root, when we feed with aluminum, or sometimes we don't need to feed aluminum because soil, acidic soil aluminum is really abundant. So we grind it into the powder, and we put one of the standard, like citric acid aluminum, or malic acid aluminum, or other aluminum, or other organic aluminum. For example, I try to understand if the catechine also combined or chelating this aluminum. Then put together, we just put into a glass wire into the MR machine, and you can give the different iron sheet. We do the aluminum 27. Yeah, we can see the sheet that when combined with the different chemicals, they will have the nuclear ribbon, the turning renaissance shift. You can see the signal from the standard compared with the standard. We know which one is the major compound. Yeah, mixture. So that's how we use. We don't do the. This is one way to utilizing the NMR, but also other people just use non-destructive, quickly can profile many different compounds. Then do the modeling, do the simulation. You will give just like a very far-read spectrometer, or you can establish a model, different chemical model, use the very accurate measurement to see this sample. Of course, this need to set up like 100 sample, more than 100 sample to set up the different the model. For example, I want to detect the seeds, soybean seeds. I don't want to destroy it. I just want to collect the seeds from the field. We have 200 different varieties. We want to detect the oil content quickly. You don't need the grind the seeds, and you just put the seeds, or even for some machine, you can do the grind into powder and put in the. I think we use before we use the far red. NMR also can do that. You just put in, then they go to very complex spectrometry, absorption or reflection, those signals. Then you also measure those compounds. Yeah, measure the inside fatty acid, flavonoid, isoflavone, or carotenoid. What is serine? See, there are five you concerned. Those metabolites, you measure those. Then use those cotton to have very, like 100, 200, or some, to do the modeling, to do simulation. Then each compound, you will find have one way, very regular change with following your compound. Your for example, fatty acid, your 18-1 oleic acid. 18-1 oleic acid, you found high oleic acid, this peak high, or low oleic acid, this peak is lower. So do simulation. Then, as long as you set up the soybean seeds detection, use this machine, IMR or far red spectrophotometry, then you can, next time, just directly, of course, this time you need to use accurate machine, SPLC or GCMS, to measure all those compounds, to do simulation, to do modeling.
Could we give 5 to 10 minutes break?
Okay.
For you?
Okay.
It will be good for you also. No? You are speaking from the line. So we take a break, and after that, yes, we comparison of the metabolic tool. We can go ahead with the expression protocol for the metabolic.
Okay. Okay.
Will be in the same slide or that?
Yeah, same.
Okay. We take a break.
Okay.
So thank you, Professor Jain, for this first lecture of this course. In this course, first lecture is related to the broad, that is the application of the metabolomics. So question come up that what is the metabolomics and what is the real useful of the metabolomics. So if you go in the previous civilization, so you know many persons can identify a diseased person. Just see the color of the skin, can see the color of the cornea, and can understand some disease in any human by knowing the color of urine. So that was something associated with some metabolites which changed during the course of the different biosynthetic pathway and the degradation pathway, and responsible some biomarkers for the different disease detections. So it was the typical biochemistry that happened 100 years before and 200 years before. So now there was a question that come up in many persons. So how these compounds are synthesized during some specific disease conditions, and how they are synthesized, what are the situations in which they are synthesized. So this was associated with the medical history of the human civilization. But if you come on the plants, so yes, plants metabolomics is just evolved in the last 10 to 20 years vigorously. Before that, most of the metabolomics was dedicated on the medical sciences, and now we are very evolved in terms of the metabolic analysis during the different disease. Even such, all the cancers cells have their specific metabolic profiling, and we are able to identify some markers which can help us to know that this cell after 2 years will be behave just like a cancer cell. So now we are on that prediction tools, and many AI is also integrated in that concept. So now, before 5 to 6 years of the occurrence of any disease, it is possible to identify a cell in the breast cancer which might be responsible for the cancer in the female. So try to understand. That's why that is the importance of this metabolomics. And if you think about the plants, so plant, every plant has some useful applications. We all the humans have the different faces, different facial features, but plant species are totally different from the others, and they have something unique that is also define the human civilization. So those are the secondary metabolites. These metabolomics can be categorized in the two different categories. First is the targeted and second is the non-targeted. So let me explain the difference between the targeted and the non-targeted. Targeted means you know very well what type of the metabolites you are going to analyze. Non-targeted, you don't know what will be the compounds you are working, you have to find out, you don't know anything about those metabolites. If you are profiling of those metabolites, means you are investigating the occurrence, their quantification, their identification, so that is labeled as a non-targeted metabolomics. So these non-targeted and the targeted metabolomics can be used by the different techniques, and the most important techniques that is almost available in many laboratories, that is the GCMS, LCMS is also available, otherwise NMR is also present in other chemistry labs, HPTLC, and if you don't have anything, so a very small tool can be used to differentiate the different metabolites, that is the TLC. And all of you performed some experiment during your graduation or before the 10 plus 2 TLC. So TLC is also tool actually, that is a part of the chromatography. All the chromatography is evolved on the TLC. And in the chromatography, that is HPTLC, HPLC, and then LCMS, capillary electrophoresis, those are pushed. So yes, the question is that, okay, we have so many machines which we can use to generate some type of the data. So I am considering the partitioning of the whole metabolomics into the three different categories. The first is that you have to extract some metabolites from any organs of the plant or animals. So extraction of the metabolites in an appropriate solvent is the key procedure for the identification and isolation and the characterization of the metabolites. The second point is the selection of a tool, which machine you want to use it. So every machine is always giving some data. Every machine is giving the data actually. Every, whatever, yes or no. So now question is come upon us that what data is meaningful, what data is genuine, how we convert those numerical values into some meaningful information. So for that we have to use some tools actually. So we try to understand those tools also as a part of the third part of the metabolomics. So whole metabolomics can be covered into the three different parts. First is the extraction protocol, because every metabolites, some are the positive charge, some are the negative, some are the neutral. So if you are using the same extraction protocol, so might be you are covering only a specific type of the metabolites. Some metabolites has the different ionic strength and they can be extracted by the different type of the solvents. So that's the second lectures, and Professor Jain will cover these second parts also. He will cover many more things related to the metabolomics, and today the schedule of the Professor Jain is really very busy because he took this second lecture, then he also took the tutorial. In the tutorial you are free actually. We speak, you ask, we give the answer also. It's of the 2 hours, 1 hour will be we take the lectures and then we do the interaction along with the metabolomics. So that is the part also. So your 2-day schedule is busy. Tomorrow I have the 2 classes, you have the only 1 class. So little bit relaxation for him because he covered 3-day traveling. So yes, I would like to invite the Professor Jain also for the second lecture of the GYAN that is associated with the extraction protocols associated with the metabolomics analysis. So yes, Professor Jain, please, you would like to invite come again and then speak about these metabolomics and other things. Yeah, I requested him to make the special type of the coffee for you, but he is a little bit late. You can take in between your lectures also. That is not an issue. Continue. But you have to cover those solvent extraction of the story of the solvent.
If you have any question, I think that learning, 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 experience, my teaching experience is long. It's like my age, but also my experience as a student, and also tell me that when we're learning, when we listen to the class, actually the more efficient way is to dialogue, not a monologue. So if you have any question, just raise your hand. Then we can stop, and also we should no need to catch up. Yeah, another point is for the machine, for the learning, we have a training that I attended many different training, like HPLC very earlier, and later on LCMS, and the USA, or even when I do the PhD study, we all need to get training and learn. Particularly when I do PhD study, we need to manipulate those. We need to manipulate this machine, because we also need to do the chemical structure and analysis, IRA or NMR, what's the amount of the sample required for the NMR analysis, then sometimes we need to depend on sensitivity, and also depend on the power, how many 800 the medium, what's the or what else. So my experience is when we study this, and also just like another professor said, probably we don't have the machine currently at the department of the university, but we can set out for very specific professional service. They can give us the right profiling. Well, we can consult with their service professional. Actually, okay, I want to analyze carotenoids, how to extract them. So we know we need to learn the carotenoids. A carotenoid is the hydrophobic. Yes, not completely dissolved in water. We have to use some chloride or mixed with other. Like too much chloride is not good. Probably we need to use the ECS ester. Yeah, use the ester evaporate very well to mix together to use a certain proportion of those solvent to the extraction. So all those, when we study the metabolite, it depends on the different. Okay.
So, actually there was a coffee a little bit late, so I would like to give him 5 minutes break. So we will take the coffee. Meantime, you take the coffee. And second is that there is a little bit introduction with all of you. So whoever is the participant comes from the outside and the inside, who are the registered in this metabolomics Gyan course. So they must give us their details, what type of the work they are doing, why they are here, and what they expect from us. So that within these 5 days, we try to meet their expectations in any way, and we try to help them whatever they expect from us. So one expectation of everyone is that this is a foreign country based from the China, and yes, what is in the China and how they are working. So that is the first question with all of your minds. Second is about this course. So how this metabolomics course. Please forget it is about the plants and the animals for things about it. Yes, we are working on the plants, but this metabolomics is uniform, is working applicable in all the different types of the organism also, including the algae, bacteria, fungi, plants, and whatever the organism. So yes, a brief introduction, only just your name, you are from where, what is your expectation from this course. That is a small 10 minutes round of the introduction. So let me start from here, and I think we have speaker or not. Is it working or not? ये काम कर रहा नहीं कर रहा? वैसा नहीं है दूसरे वाला. Second. Yeah. Is it working or not? So, I would like to start from the Dr. Jyoti ma'am. Dr. Jyoti is a faculty in CDLU and recently we met and she was very eagerly waiting for some interaction with the Department of Botany, and within the 5 to 6 days she is here, so we can appreciate her zeal and affection to do the collaboration as well as to interact with the this foreign faculty and about this course. So yes ma'am.
Thank you so much, Dr. Vinay Yadav, to providing such a great opportunity to everyone and providing this platform where we share our experience. And I'm also highly thankful to Dr. Zhao to come here and share his expertise with us. So I'm Dr. Jyoti from Chaudhary Devi Lal University, Sirsa, and we are also working on the medicinal plant which used by native peoples and used to cure various diseases. We are also working on their metabolic profiling and also nanoparticles synthesized from these medicinal plants and how we use these nanoparticles to treat various diseases. Mainly we working on the anti-cancerous properties and anti-diabetic properties. Thank you so much, everyone.
Very good morning, Professor Jain, Dr. Vinay. I'm Dr. Anita Patil and I'm a senior professor at the Department of Biotechnology, St. Gargi Baba Amravati University. My area of specialization is plant secondary metabolite, and I do have a lot of publications in the secondary metabolite, about 10 to 12 research projects and 7 granted patents. Recently, I have got a major research project from Government of Maharashtra, about 7.5 crores. In that, we have to develop certain drugs from the plants. One of my patent has been already commercialized for a kidney stone, and that has been marketed by a company in India. So results are very good, and it's a very promising area of research, because when I started doing research on plants, everybody used to laugh at me, and everybody used to say that this is not a research area. Now, Government of India has already sanctioned Ayurvedic biotechnology is one of the subjects in UGC net also. So, dear students, it's really a good area of research. Everybody has to work it because we have to follow some tradition of India also. Okay, and I have seen in China, Japan, everybody, you are having very much belief on plants. We have lost that belief in the middle of that, and now we are again having belief on the plant drugs. Yeah, thank you so much.
So because we have only 5 minutes, so this introduction is only from the faculty. Rest of the participants will be invited in the next class afterwards.
Hello, good morning, good afternoon everyone. My name is Pushpinਦਰ Kaur. I am assistant professor in chemistry department from Shri Gobind Singh College, Chandigarh, which is affiliated to Punjab University, Chandigarh. And my specialization, I did my PhD in natural products, and I'm working in plants secondary metabolites like the isolation and the characterization of the secondary metabolites from the medicinal plants. Another area in which I'm working is the nanomaterials, like I am synthesizing the covalent organic frameworks for different applications, like for the catalysis, for drug delivery, and for the sensing applications. So how I got interested in the metabolomics is that in 2013, when I was doing my postdoc at Impact Chandigarh, then I got to know about the metabolomics, and I really got fascinated by this area. But that time I did not have any guidance. I just read the papers and I really wanted to work in this area. Then throughout my journey doing another research works, this was the area which was kept in my heart that I want to work in this area. One more reason for this area is that what I found that this metabolomics needs limited resources. Like I am working in a college, so we have to survive in limited resources. So more of the analysis part is there. Experimental is comparatively lesser as compared to the like my other work, like the synthesis and the other things. So that is also I am interested when I shared the brochure in one of our groups, then I really got interested that now I have someone to guide me in this area. So that's why I'm here to learn from Professor Jha. Thank you.
Yeah, ma'am is my senior in CSR HVD Palampur, so she also did the PhD from there. Yeah, and Dr. Atif, I just want to introduce all of you. He worked with me in the NIPGR, and he was the first person who made a call when I shared this brochure, and he made a call from the third one. I would like to join this metabolomics. So yes, faculties are the less, but whoever is sitting here, they have really great zeal to pursue their careers and give the vision to their laboratories, because this is something, as ma'am told now, it requires lesser resources. You just need to prepare the sample. You take some very good machine from somewhere who can operate very precisely. You just get the raw data, analyze yourself, and you can easily get the paper. But your experiment must have some biology in that experiments. Otherwise, there is no meaning of the data. So, Dr. Atis is with you.
Yeah, thank you for inviting Vinay sir and Professor Zian. Thank you for coming here. Myself, Dr. Atish Sardar. I am assistant professor and head of the department in Jogesh Chandra Chaudhary College, Kolkata, affiliated to University of Calcutta. So I am working on arsenic accumulation in rice, but most of the people all over the world and also in India, they are interested in high yielding rice varieties. But I am interested in our traditional rice varieties, which are aromatic rice varieties. And in Bengal, before independence, there were 100 aromatic rice varieties that were used to be cultivated. So we are trying to revive those varieties, and their fragrance and their taste are, I can vouch for that they are better than basmati, which is the best in the world, which is considered the best in the world. They are better than that. We need to introduce them. So I want to know the biology behind why these aromatic rice are aromatic in nature. There are preliminary works there, but I want to know the details, and I want to go deep into that. That's why I am trying in this new horizon. I am not an expert. I am just came here to learn. Thank you.
So yes, we would like to invite again Professor Jian to start the extraction protocol. Extraction protocol is something that is the key starting points of the metabolomics. The first question in your mind, please come sir. What metabolites you need to identify? What metabolites you need to investigate? Then according to that you have to identify the protocol.
We have compared with the different analytical tools for the metabolomics, analyzing the NMR, this different UPLC or HPLC. This is for separation. You just know it from. Become a knowledge on UPLC, the faster and higher resolution. And HPLC is very common. Right now it's still very common, but their performance is different. The mechanism is also the kind of mainly the column packing, the particle, the size is different. The size is smaller, then separation is higher. The separation efficiency is higher. Increased separation definitely will decrease the size definitely will retard the solution going through. That's why we have to use the high pressure to press those to go faster. Otherwise, we won't just like a particle more smaller, they will become gel, right? Become very sticky. So that's why they use the high pressure, we call the ultra pressure or ultra performers. The reason is like this. This is the metabolite analysis, but generally, this is a very general metabolite analysis. Before if we come to the metabolite, this is LCMS or GCMS. We know that the GCMS analyze volatile, right? The LCMS analyze non-volatile. Yeah, non-volatile sometimes this really depends. The GCMS recently also can analyze a lot of lipid, like a cuticular wax. We know every plant, most plants covered with the cuticular plant on leaf surface or stem surface. Those cuticular wax, the white, the very shiny one, we looks leaf, right? On the leaf surface, the cuticular wax. Cuticular. Yeah, cuticular wax. But anyway, those shiny stuff is like the long chains fatty acid and together with the other multiple components like glycerol and also sometimes saponin or sometimes the other hydroxy fatty acid, hydroxy branched fatty acid. So analyze this is another thing. So analyze those compound. Yeah, some people say that we can use the LCMS. Actually, the GCMS is the most widely used. How they do that with lipid, with just like our candy, the candle, right? We're lighting in the room. So we have to do the sample preparation for the utilization, derivitization. We have to treat it with a certain common fluoride or silicon compound and try to treat those wax with react with the chemicals. After reaction with chemicals, then we purify, then inject into the GCMS. So this is also the one type of, it really depends on the different chemical, right? When you analyze fatty acid, you can use the GCMS. You can use the GCMS, but you also can use the LCMS. No any problem. LCMS can isolate different lengths of fatty acid. But use the GCMS is very common because very mature, very sophisticated the method after extraction, then react with the methane. React with the methane under the alkaline condition. We usually add sodium, hydroxy sodium. Yeah, under those conditions, the fatty acid quickly, methylated. So after we load on the HPLC, then we extract again, load on the GCMS, then we can get the methylated fatty acid. The 18-1, 18-16-1, like normally we observe the plant fatty acid. This is the comparison of the different analysis tools. I mean, like some of them very highly sensitive, you can see very deep details under sea in iceberg. But some of them, like only can get so big you can see, like MMR. That's one of this strategy to analyze the metabolite carbohydrate, we use the GCMS, HPLC, LCMS, HPLC, we need to couple with different type of detector because the carbon dioxide, different sugars, they don't have absorption, right? They don't have the chrome, so they don't have absorption, but we can use their reflection, the solution through the pattern, through the reflection detector, we can detect different carbohydrate, different sugars on MR, and also the lipid use the GC-MS and also use the LC-ESI, electrospray ionization. This later on we will talk about how the mass spectra works. Just because after injection in the intersurface, there are several types of ionization. All the sample, if you go to the mass spectrometry, you have to ionize it. Because there are electromagnetic field need those particles have to be charged, then pass through the electric field, then separation according to the molecular mass, molecular weight, and also charge degree. The ratio depends like how much finally in the detection, or somehow it goes through the trap, ion trap. They can filter some part of some type of ions, so only detect part of it. But later on we'll talk about like a Q-top. The Q-top, LC-ESI top time of flight mass spectrometry. This is the most commonly used for the metabolite quantitatively to metabolite profiling. Usually we use the LC-ESI Q-top MS. But for others like Q-trap or tri-drop or quadra-drop, those mass, you only can detect a certain type of molecular. Those molecular can be very accurately quantitatively determine the structure. So those are different purposes. But for metabolite profiling, usually use LC-ESI top MS. The amino acids usually we use we also use GCMS or OS, LCMS, but also we can use the amino analyzer. Those pretty common for the protein. For soybean, we need the analyzer, or for some others, the nutrients very significantly reduce our nutrients, the quality. The vitamin usually use the LCMS. Recently, there are many different protocols have been developed to analyze the hormone. Also, they use the LC-ESI Q-top MS. They try to actually measure the different type of hormone, like jasmonate, like the oxalipine, the hormone. They can measure jasmonate, and leucine contribute, or OPB, those precursor for the jasmonate biosynthesis, and also methyl jasmonate. They all can analyze with the LCMS, different type of. Also for auxin, they can analyze IA, IDA, or others. For cytokinin, they also have different type of small, those purine compound. So for those, for example, lipid, definitely we know lipid is not dissolved in water. So we have to use the chloride as a major component, and sometimes we need that as a method. So for lipid, for every lipid profiling, we can, there's some lab, like Kansas State University. They have a lipidomics center. On their website, they have a protocol how to extract the plant lipid. The neutral lipid, like a glycerol lipid, or the polar lipid, like phospholipid. Phospholipid, the structure, they have a two long fatty acid chain. Then in glycerol, the backbone. Another third hydroxy to connect with organic compound like choline. If choline, those lipid called phosphocholine. If the inositol, they call the phosphorinositol. If they call the acetylamine, they call the acetylαμine. It's a different kind of membrane lipid, right? They have polar just because they polar. So the lipid, if we put in the membrane, they will self-form membrane. That's why synthesis membrane people do the transporter assay or do transmembrane protein structure. They can use the synthesis just with different ratio of TCP, DI, and TS serine, phosphoric serine. So those during the extraction, they have a tips. For example, need to prevent the oxidation because those phospholipids are very easy to oxidize. During the extraction, we always need to care about the oxidation because the chemical. For this, the reason why we need to care about the extraction is because during the chemical identification of many authors, scientists published the structure, said this is natural product, natural structure. But actually, later on, they found that this is artificial. Why artificial? Because during the extraction, some chemical reaction happen. The chemical reaction occurred cause the new compound generate. The new compound, we think this should be natural compound, but actually not. People use a different way to extract, use a more natural, more productive way. For example, when we extract for LCMS, we usually use 80% of methanol. So those, you really don't need to write down something. You don't need it. Just to understand the key point is that we want to prevent oxidation. All the lipid extraction, we need to use one of synthetic the chemical PHMS. I forgot which the chemical, but the abbreviation. The chemical is widely used for food industry. This is safe. Food industry, as long as you call them the oil or your fried products or something, you try to increase the shelf life of your food products. Usually, you add this compound. This is regarded as safe. Also, in lipid extraction, we also use this compound. Need added to the solvent. Need pre-added solvent. This is like oil solved the chemicals, just like vitamin E. Vitamin E is oil soluble antioxidant, but vitamin C is water soluble antioxidant. So sometimes we extract proteins, we need to add vitamin C, or we analyze the other extraction, we need to use antioxidant. Prevent oxidation. This is one of very important. For lipid extraction also is very common. We need to add the lipid. But for amino acid, we know amino acid is very stable. Amino acid is stable. So normally you don't need to add those. However, for vitamin C, for vitamins, this depends. Vitamin C, you can itself is an antioxidant. That means itself very easily to be oxidized. We eat vitamin C just because vitamin C, when we take in, they can, as an oxygen species, but themselves is saturated into the oxidase vitamin C. They themselves being oxidized, but protect our biological molecule of RNA, DNA, protein, or other sugars to prevent. That's how they work. So we add those antioxidants to the lipid extraction. There are always some protocol. I did not really prepare how the extraction is, what's the proportional to chloride or methanol, or it really depends. For example, carotenoids, you can add some chloride, but not so many. You also, because this large molecule, you add some ester, the SL ester or ECL ester. How many carbons really people really do the analytical method protocol? They can develop, they can compare which one is better. We just take their reference. Most important, we know what tips we should take care of. We should pay attention to prevent the oxidation or to prevent other artificial compound generation during the extraction process, overheating, or the light, some light sensitive, for example, acetylene. We know acetylene doesn't matter is delphinidine or cyanine or the peladonidine, all the light sensitive. So we usually use blank paper or aluminum foil to cover those plates, to cover these vials. Even we do the sonication. Usually sonication is one of the common ways. But even do sonication, we in water bath, we put some ice into the bath. Then we put our vials for 1 ml, or you use large scale extraction. You can use to surface and then sonication. Usually sonication is the most effective way to quickly degas. Let the solvent quickly penetrate into the tissue and then extract. So those also the fresh tissue or dry tissue matters. A different tissue type. Then you are determining that if it is dry, or because the tissue contains some water. No. So this need you consider. However, in model, in most many scientific research, we only do the comparison. We do molecular biology. We only do comparison. We don't do absolute analytical paper. We don't want to publish the analytical chemistry. This very high profile journal. The specifically mentioned all we use this develop this new tools to analyze. Do comparison. You just compare the different fresh tissue because you concern the molecule biology, right? Those molecules related to the enzyme or same tissue. Usually when we do, for example, we try to do the metabolite profiling. We also use the same tissue, mixed tissue, or individually repeated from different plants, biologically repeated. From 6 plants or from 5 plants, you can see, okay, these 2 plant tissue are mixed, seeds are mixed, whatever. And one biological repeat, another biological totally we can take like 6 or 5, but 3 samples, because when we send out or even analyze by ourselves, the kind of abundant, time consuming the stuff. So usually we do 3 repeats, but the 3 we can represent more than 6 biological repeat individual plant. So that's to make sure your sample, your result is meaningful. After that, you mix it, you try to do the metabolite profiling. Okay, just take some mix to the metabolite. But the other part, you can send out China, if you see the many publications, many of them just do this kind of way, do work in this way. One part analyze, take the sample, fresh tissue, they grind it into liquid nitrogen, then into powder, then some send for the RNA-seq, and some send for or even others, like do the accessibility. This is completely developed from a 3D genome. The people think the strategy of behind the mechanism is they think if I do the accessibility, no matter what kind of sample, in certain time or in certain situation, the RNA, we can, the transcriptome we not only get from the result, is how many transcription relative abundance to see, to explain that, and also from another way, from accessibility, you can understand with the reference genome already sequence there. You just check, use the AC, those kind of way to check 3D from 3D genome angle to understand some gene in this tissue or under this conditions, they more easily to be activated for transcription and then translate into the protein to change the metabolite process. So anyway, you can do the as more as possible in the fresh tissue. That's what I said is depend on your purpose. If you want to quickly understand the metabolite in different tissue, do a comparable study, and also try to understand underlying the molecular mechanism, then this way is the option, the best option. So you don't need to dry it tissue, because the drying process will probably also change something, right? So that's why people just okay, just use that. But this depend on do your metabolite dissolve in 80% of methanol? 80% methanol is widely used for the non-targeted LCMS takeoff, the metabolite analysis. This needs another consideration. Should I use the fresh tissue? I add some methanol to mix with to make it compatible with water inside the tissue water. I also add some chloroform. Then probably also the acetone. Yeah, acetone is also very good solvent. So in this way, I can extract much more the non-water soluble metabolite. So my preference metabolite change for the non-water soluble part. So you will see the abundance of the carotenoids or some lipid, fatty acid or lipid dramatically increased in that MS, because in later on the Q-top MS already the barrier rate from already I think to the 2000 molecular weight already to the 2000. Before usually we limited 1000, right? But now the capability already increased dramatically. So like 2000, many large saponin, like a 3-ring or 4-ring saponin, the glucoside or several glucoside attached, we can analyze very well. So this kind of extraction, we don't do that unless you do the analytical control. You try to purify the compound. For example, the professor wants to understand what compound in this medicine plant can prevent, can change Okay, in China, I know the one of our colleagues, he just tried to study enzyme. And if you take the enzyme, somehow they can to dissolve the crystal of the calcium, carbon dioxide, and calcium, or the others, the crystal. It can dissolve.
COD and COM.
Yeah. So this, and then also another way to study the kidney stone is people think from maybe they need to adjust. They take the medicine long term, and then the whole body adjusted. So probably not really the indirectly effect. But anyway, I mean, sometimes when we do the bioactive compound isolation or targeting in this particular medicinal plant, usually we extract both polar, non-polar part. Then to check, if you don't have the medical model, you don't have biomedical model. For example, you set up a model. I use this extract quickly test if this portion is effective or not effective. If you have this way, that will be great. But if you don't have this way, so tracking those bioactive proportion, finally to isolate the target compound will be very difficult. Probably multiple compound, like traditional Chinese medicine mixes several different plants apart. They call them multiple targeted, but actually for the Western country medicine, they don't believe those. The multiple target means to say nothing. Right? They need a chemical clearly structure identified and the target identified, and then the pharmacokinetics, the pharmaceutical dynamics in body and the toxicity, all of those need confirmed. You don't have side effect. If your toxicity is higher than effectiveness, then the problem. So those all those when we study the bioactive compound, considering about which proportion, which extract part need to consider. I think need to be tractable. For amino acid, hormone, and hormone also very easy to. For example, we isolate the IA. We want to isolate IA. We want to care about the growth effect, or cell elongation, or some effect, or root. Then they need to have special tools. We have to avoid the light. We have to put on the ice all the sample. Then try as more as possible put on the ice. And also in extraction need to be in the dark. Yeah, because the light, we know the IA or ADA all is light sensitive. And also others, plant hormone too. So the organic acid you also can use aromatization to do the methylation or use the BAFS, those chemicals to siliconization or methylation or some treatment. That's them easily to be gasified, to become gas. Because usually gas spectrum needs like 200 or 250 degrees to evaporate those your sample. Evaporate the sample, then quickly go through the very long the separation column. Then eventually go to the mass. Those particles also need to be of course, this the first somehow we use the inflame to detection, or some use the mass spectrum to detect. one of the sample, of course, later we can talk, like a lipid analyzer, need to use the different solvent, prepare sample, need to be charged. For example, use the ammonium in the lipid, phospholipid, or the neutral lipid, like glycerol or sugar, galactolipid. Galactolipid is much more enriched in the photosynthesis of the chloroplast. Chloroplast membrane, most of them enriched with galactolipid, like EGBG, all those components occupy like 80%. And many jasmonic acid also, we know jasmonic acid is generated in chloroplast, right? Yeah, in chloroplast. Yeah, just because those photosynthesis specific lipid, if as long as they have 18-3 long chain fatty acid, they can be enzyme catalyzed jasmonic biosynthesis in the chloroplast. But those long chain fatty acid either come from EGBG or MGBG, the galactolipid. The galactolipid also is a non-polar. We also need extraction and use the specific. After extraction, before sampling, usually we use both the sodium chloride, no, sodium hydroxy sodium to ionize those lipids. So after ionize the lipid, you will see the average molecule when added with sodium. But for another negative charge, for positively charged, we need to add the ammonia. We use the 5 millimolar ammonium to mix it with the lipid, mix it together, then do the injection. Use the ESI top MS. This I think this is from Kansas State University website. They have a lipidomics center. Normally, because they have this is my previous supervisor when I studied in USA. So they have established very the world-known lipidomics center, analyze both plant and animal sample, very accurate, large amounts of phospholipid, galactolipid, or neutral lipid in glycerol or DAT or TAT, and MAT, the mono glycerol, and mono glycerol. So all those, I mean, need to depend on the sample preparation. I guess many of you don't work on the cation or ion. So we work on the ion or cation because I'm trying to my project is study the food safety. We're concerned about the aluminum or heavy metal, all those even anion, like fluoride, chloride, or other even organic anion. How they accumulate in plants, in tea leaves, so affect our tea, the safety issue. So we also do those kind of analysis. This is I think for organic acid also, all of those extraction, unless you do the analytical purpose, try to develop a new method or do others, usually do the target or non-target really depend on what's your purpose? So extraction also depends on your purpose. If you prefer to analyze the compound, or you know the target compound, the hydrophilic or hydrophobic, those, then you can change the extraction ratio. Yeah. For example, we once studied I once studied the huge caterpillar. I call it caterpillar wax, but anyway, the wax is very important to our plant, not only for the protection of plant from the pathogen, but also actually in tea processing. Those very thick caterpillar wax can be degraded. Honestly, because some of the, we know the caffeine also accumulates in the surface. Caffeine also separated from ectodermal cells, synthesis in the probably chlorophyll cells, then separated into surface cells, probably continue separated to the leaf surface. So you can very easily, initially when we first extracted to the GCMS, we found this caffeine, the huge amounts of caffeine. We thought this is contamination, but then we used some other tools. When we measured some other tools, use the galectin. The galectin protein, like a gel protein. They use the galectin to make a membrane, then put it on the leaf surface. Put it on leaf surface, then stick, then lift. For a very short time, then lift. Then the galectin will remove the caterpillar wax. They don't remove others. Initially, we thought that probably when we extract the caterpillar wax, just take the one leaf, then quickly put into chloroform for 30 seconds. Or longer, you can do 1 minute, but no more than 1 minute. Otherwise, it will destroy the inside cell. People will ask, you have contaminated with the cell internal metabolite, not the surface, not the extra cellular metabolite. So all those, after you use the whole merge the whole leaf into the chloroform. So 30 seconds or 1 minute, then you leave it. Then you just dry the chloride, dry chloride, then do the BASF or another chemicals, do the silicon or derivatization reaction. After that, you can quickly apply it on GCMS. So that's kind of difficult. Sometimes you couldn't get anything. And this caterpillar wax also can easily oxidize. You cannot, after extraction, put on the even refrigerator -20. It cannot be too long. You have to analyze within 2 days. Put on the load on GCMS, then analyze the different compound. So we can easily observe the caffeine. Indeed, on the metal we use the solvent, chloroform merge method, or use the galectin to remove the surface appeal of the chloro. Because protein doesn't contain others, right? So we use the protein quickly, then use the solvent to remove the stripped caterpillar wax into the solvent. Then we try to do the same. Both methods show the caffeine, and so the caffeine on the surface, leaf surface. They're very interesting. But anyway, those results have been published. But the similar thing actually happened in the in American in the North South American Argentina, they have their plant called the tea called metabolomic. The Argentina drink a special tea called what's made?
Yeah, Lionel Messi drinks that.
Yeah.
I'm forgetting the name, but there is it is bitter in taste. It is quite.
Yes. But also that plant, just like a tea plant, they contain a very high level of caffeine. So that's why all the natural the drink they contain high level of caffeine. Otherwise, probably people don't want to drink.
That is yerba mate.
Yeah, yerba mate. Yeah, I just recently reviewed their Amber journal. Yeah, I think Amber journal. Yeah, they submit the genome sequence for the yerba mate sequence. That's very interesting. People study the.
So should we take the break for the lunch right now?
Huh?
It's a lunch time.
Okay.
So should we take a break for the 1 hour? So now we are declaring the lunch time, and whoever is a participant, they are invited for the lunch at the guest house, and we try to gather here in the 131 right now at the first floor, just adjacent to the first floor. There is some NIRF chairman actually. Professor Tulsi will be here, and so we have to sit our lecture of only 2 days from this seminar hall to the 131, that is on the first floor. I will guide all of you. And in the morning, every day we have the lunch here from the morning up to the evening. And what is the Gyan mandatory things is that every day at least 4 hours teaching. And please take it a note that at the end of the day, we will also conduct a small test, and we will also give the certificates on the basis of you, whatever you will achieve, your get the marks actually. So this is a Gyan course. It is the 1 credit, and this credit is useful for the faculty for their professional development as well as their career gas also, and for the PhD students, this is just like another credit they gain during their academic achievements. And for the MSc students, yes, this Gyan course is equivalent to the 1 credit. If you some way have missing some 1 credit, so you can use this credit for that purposes also. And yes, Professor Jain is just accommodating. Now you are able to listen what he is telling you, because at the morning there was a gap for the communication from our sides also and from his side also, but now I think things are clear. So you will retain very good knowledge here, and just try to understand one PowerPoint has enough strength to speak for the 1 hour. So if we have the 20 slides and they are enough to explain the whole concept also. So we will gather all after 1 hour at the first floor in the 131 only for the participant right now in the evening session, because it is the tutorial. There is a need to interaction with the speaker also, with us also, as well as to show your problems, how those, whatever the topic, how those topics will help you also. So that type of the things will happen for the after lunch. So yes, please for the lunch, proceed for the lunch. So, go ahead with the metabolomics, especially with respect to the spectroscopy. So Professor Jain, please come and start. Applicants will come very soon actually. Start it. Otherwise हम शाम को लेट हो जाएंगे.
Good afternoon. So I will continue the course presentation. So in the morning we already talked about some information about the metabolomics analysis. The one point we have said the LCMS or TCMS, the liquid chromatic coupled to the mass spectrometry, and also gas spectrometry coupled with the mass spectrometry. Both are most commonly used for metabolite profiling. So both one is for volatile, the most often like TCMS. Another is for the targeted or non-targeted non-volatile metabolite profiling. However, because the TCMS and the LCMS have very different way to processing data. Therefore, many people actually like TC to profile, to do the metabolite profiling. As long as your target metabolite can be volatilization and it can be gasified, can be evaporated. Otherwise, you cannot. However, so far, even like supposing we still can do the mass, we still can do the volatilization and do the gas TCMS analysis. So the TC become now more and more powerful and use more widely. Also because the LCMS data process is very difficult, very complicated. Now LCMS doesn't matter. You set up the liquid profiling or non-polar liquid or liquid. Because my previous terms with Dr. Roosevelt, who is now in charge of Kansas State University, the lipidomics center. She is in charge, female professor, very great professor. So she is very familiar with the mass spectrum, particularly LCMS spectrum. She is very good at identifying the peak. According to later on, we will talk about how to analyze the data through the peak, ion peak, the molecular, the mass, molecular weight to the electron to charge ratio, identify how this molecular weight represents. Usually there are some rules, the different type of compound, like flavonoid you often see, because the way you analysis mass spectral data is you understand how they generate the data. The first ionization, then the generator, then need to use the neutron to break it down. To break down the mass part is that break down the molecule, broken into small pieces. So the original molecule we call the mother molecule, and it generates the two pieces, broken down pieces, we call the daughter pieces. The daughter pieces, the molecular weight added together should be equal to the mother, the daughter mother, not father. Mother molecule, the molecular weight. So according to this calculation, later I will tell you how we get the mass spectrum, pattern mass spectrum, two mass linked together. The first ion peak, ion broken down. The broken down, as long as they qualify for some secondary analysis, then we will collect, then set for the second mass, then broken down again. That's way you can analyze very completely the molecule, determine what type of molecule. At least, like flavonoid, we usually can see 273 molecule and iron peak. The very standard, like marker. As long as you see 278 or 273, this is the flavonoid. Definitely, you look at the original, the mother you can find that. So then we will use our piece to explain how we calculate, how we track this. However, in GCMS, usually we have a very good library. One called different like terpenoid, or you use fatty acid, oxalating fatty acid derivatives. You know, we know that volatile usually in lab, volatile compound can be classified into several different classes. One class is terpenoid. Terpenoid usually monoprotein, satisfactory, or those half, like carbon 5, terpene, like a basic, basic backbone. Terpenoid by synthesis, you throw the carbon 5 malonic acid, then together form the IDT or MDTP. Then use the hand to peel to hand those different connection from different type of generate different type of terpenoid from monoterpene with carbon 10 to saturated terpene carbon 15, diterpene carbon 20, all those. And so on. You can generate carotenoid, and even there are some derivatives that generate other forms like BR, racinoid, and also the ADA. Carotenoid pathway, or those we call the high molecule terpenoid pathway, pretty complicated, generates a lot of important compounds. But anyway, what I mean is the mass spectra from here, the liquid chromatography coupled to the mass spectra, to the Casper chromatography coupled to mass spectra, the separate people start to have their own different preference. So, but anyway, so if we consider, we will do the mass spectra, the sample preparation. The morning we talk about the general consideration, the mass spectra. You want to look at the concentrated, analyte. You want to analyze the compound and remove the other compound. You don't want to get into cause the background ion or suppress other ionation. So this is very important, like salt. Salt, when we do the ionization, ESI, or they will suppress the ionization. So we have to dissolve to reduce the sodium, potassium, those compounds. Usually in natural spray ionization, there are some organisms in GCMS ionization is different. We call it electro ionization rate. But in LCMS there are other different types of ways. But anyway, you get the plant sample and grind it as mentioned. Remember, we need during grinding we should keep it on ice, or just we need to add some antioxidant in the extraction buffer to minimize the structure change, oxidation, other things happen. So then with the sample, usually we put liquid nitrogen, and we also, the sample is taken. Actually, this one cannot be the fluid, but actually there are fluid augmented nitrogen. On liquid nitrogen, you put in liquid nitrogen, now put the machine, they will grind in the liquid nitrogen, so we make sure it doesn't change the structure. Then you measure it, then allow that you add a solution, that will be okay. This new solution, we should have something stabilized and antioxidant or this universal. And if you want to extract some oil or oily compound, oil salt compound, then you add another compound. Actually, we use it in our food, in oil food production. So all those, then you do the certification and do the membrane change, and sometimes we need to do the pre-column. Yeah, before put you load down the LCMS or GCMS, we have to separate. But this normally you don't apply to the non-target. Non-target means you try to analyze as more as possible metabolite. Yeah, and more metabolite as possible. So you don't do the column treatment. But for analyze specific compound, like you analyze seeds, oil seeds, some secondary metabolite, you often being bothered by, being troubled by too many starch, too many oil, and you cannot analyze the seeds, the secondary metabolite, properly. So you have to do pre-treatment. That's almost no one can solve this when you grab the seeds into the sticky things, the starch and the oil, the fatty acid, the liquid mixed together makes the secondary metabolite very difficult to remove. That's why you have to do pre-treatment. That's the most often people see, because we know the seeds important. We want to know the seeds, what are, except for the starch or oil or other nutrient. What's the other small molecule with potential health benefit or potential the toxicity? We want to know this, but when we analyze it, normally will be interfere with those. So those needed, you don't need to do the non-target. You try to purify. You try to find some other small molecule. Or you don't want to analyze oil, larger, or starch, those polysaccharide, or some other larger biomolecule polysaccharide also. So the sample, I think later we will also mention, along with sometimes we know mention to the certain type of metabolite, we will also mention to different method. So you can, in the specific situation or in some cases, we can tell those sample preparation more specifically. This different type of different supply of UPLC, Q-top or UPLC, the core drop. This is the executive class of orbitrap mass spectrum. It's very different. Like this, the agilent triple core drop. Those basically bottom two, for the quality, for qualitative determine the molecular type, very accurately determine the structure, what type of molecule. At the other top, you can do the metabolite profiling. You can do the quantification very good and very large scale because the mechanism or their principle is different. Like Q-top, you can one time, after the time of flying in the electric field, those molecules at the same time affect in the detector. But in the Q-top or other triple drop MS, so they were selected. They have a selection so that you can specifically analyze certain type of compound more accurately to determine the structure. So the different purpose with different mechanism. Again, it's very general MS. I think later we will also briefly, not too much, too deeply understand this, because this, I think, just one time you cannot really understand. The major part is the first part is separation, right? HPLC separation. Just try to separate as more as accurate as possible. But honestly, only HPLC, one phase HPLC couldn't do that. That's why later I will tell you there are some other 2D variant HPLC coupled to the MS, so that you can, just like a 2D variant, the gel, the protein gel. After this divariant, first divariant, second divariant, you do like this, you can separate the protein dots very precisely. Yeah, over try as much possible, try to find the keyword, the single, the protein dot. So the basic is also the principle also that after the interface sample injection, they will have electric spray, the ESA or APCA, the spray, so the particle will be charged and evaporate into very small particle, then go to the electric, this is the electric field, then according to the different molecular weight and also charged ion, then go to, this is called Q1, Q2, Q3, then to the detector. But during this, there are some of them will be missed. In cutoff or triple top, they will be missed. But in Q-top, in Q-top, they don't. They collect all of those. And also the powerful, just because they have long electron field flying, they can separate very well, so they can detect. Eventually, they have a different time to detect. According to the time the peak show up, reach the low, about both separation and the molecule detection. Yeah, so this is different. So basically, the LC-MS doesn't matter, not only the LC-MS, the mass is doesn't matter in the front, is the LC or gas meter. Later we will see. The interface is the LC, okay. If for TC, interface is TC. Gas batteries use the circle long, the wire to separate those the gas lines in compound. Then to the mass spectrum, then detector extraction. And the detector, the extraction analyzed from the solvent evaporation or ionization. Then in detail, LC or TC just for separation or mixture analyze. The interface is for separation of the analyte from the solvent. MS analyzer is here separation and analyze the molecule, the ion, and fragmentation according to the mass to the charge, the ratio. So this is basically the principle. For example, just like what I told you, in the corner, the filter, the separate ion, based on their mass to charge. In Q-top also, in similar principle. But their time flying during the long-term flying, they are different. So this reaches the detector in different time, so that their separation, you can see the separation not mixed together in the detector, the skin. So they will have a separation effect. With the triple cutoff configuration, the first cutoff isolation, the elite molecule, the second cutoff fragment select the molecule. They have cutoffs like have ion trap. After select the summer, the ion will continue fly to the third, the cutoff selectively isolated from ion. Then for the detection or measurement. They also can do some certain measurement, but this measurement already because have some missing. So they don't have that accuracy. But definitely for the quantity, for the quality, what's the molecule weight, they will be very accurate. protect the ion or fragment, which is why the fragment, because the large molecule can be broken down to the small molecule. We still have we can cover the mass, capital mass to make sure. Particularly some lab, like a U.S. some national lab, try to analyze much larger molecule. Usually they couple like 3 or 4 mass structure together to make the ion broken down to smaller. So finally determining the structure. All of the mass structure, the analyzer through the plotting the different mass to charge ratio against their balance. That's what we usually see mass structure. The x-axis marked with M ratio Z, right? The y-axis marked with a balance. Means the peak high or taller or smaller or lower. So they find the quantity. About the ionization, the electron spray ionization is the most widely used way to ionization. In the first interface. This is just like a morning. I told you, if we analyze lipid, doesn't matter the phospholipid, those polylipid, our neutral lipid like glycerol, our MAT, MAT, mono, our diacylglycerol, our triacylglycerol, all those neutral lipid, and plus some galactolipid. That's usually analyze plant lipid. Definitely, you will guess those. Usually, we saw in plant root don't have too much galactolipid, but actually, recent years people see some plants like the legume plant, the nodule have a lot of galactolipid. So we still don't. There are some people consider why galactolipid. Actually, I have some several 2 or 3 plant journal paper talk about the soybean or mechanical transfer. Yes, root nodule. We did the lipid profiling, both polylipid, phospholipid, and also neutral lipid glycerol. And we try to analyze those lipids because one of the nature paper actually a long time ago, people already suspect that rhizobial, attack the soybean root and generate a nodule, right? Yeah, in the root hair. They enter in the root hair, follow the root hair direction, and go to the root, the calyx. In the calyx, there are some the molecule, some gene, SCR, or some were activated by the lobule. The activities then start the cell division. But anyway, those processes. But during the nodule growing, our inside their lobule also amplified to start the mature nodule and start do the same biosynthesis and nitrogen fixation. During the process, the medicine happened. People consider lipid. They found this lipid play important role. However, they have no evidence before. But just a couple of years, several years, two labs from the UK and from China also suggested that monoacylglycerol is a very important carbon supply to the rhizobium. And rhizobium use the plant, use the root provided the monoacylglycerol as carbon. So this is the first time to demonstrate that fatty acid or lipid actually very important. It actually can be used as carbon. If you cut those, the rhizobium don't grow, and also vascular tissue don't grow. So that's why we use those. But anyway, I mean, there's some those kind of electroactive ESI top mass spectral analyze the phospholipid or mesylipid very sophisticated in Kansas State University, and also later in China also they have purchased those kind of whole set of machines to set up the lipid analyze very well. And this one is not very used very often, but most commonly used is electrospray ionization. So for the analyzer, we have already repeated, but the difference is the Q-top, there's no stational or mobile phase. The separation depends on the kinetic energy, the velocity, the speed of ion. The ion hits the detector at the same time and at different time, and also have different speed. So they have long enough the tracking, flying time to reach the good separation. Ion trap and also follow up, they are different LCMS. I think the professor will talk about more. So I will just quickly go. LCMS, the combined separation, very great separation power of HPLC UTLC, then with the detection power of mass spectrometry, is used different interface. One interface is the ionization, another is the HCI to eliminate it. But in TCMS, we use the electron ionization to eliminate the solvent as generates the gas phase that transfer to the opticals of mass spectra. The interface classification different ionization method, and those ionization method determine the class of substance available for measurement. Actually, this also is the one consideration we want to use which type of metabolite. Not only consider the data analysis easier or difficult. For the multi high throughout metabolite profiling technique, definitely we use ESI top MS. But for the molecule, for accurate structure determination, you want to use like exactly what you draw, Kelvin trap, those kind of more accurate structure analyzer. But all those sometimes use ESI, most often we use ESI. Some of them use HCI. Electrospray is a really great achievement. Significantly increase the mass spectral performance and the power. Later on, they increase the energy of the detector and also the electric field, the hull make it longer, and the Q-top fly longer, so that's really increase the powerful and accurate. So this tool, I guess many of you probably agree this is just the engineer, no any high ranking, no, but suddenly gets the Nobel Prize for his a couple of years ago. He accidentally used different chemicals, but generate very good the protein. Actually, he used the mass spectra to isolate protein from some the chemical actually. Then analyze ionization, how important, how helpful. But I mean, it's just like this story just hits one truth is that any significant finding really depend on your focus, on you always consider and try different way, and suddenly the thing happen. And if you don't realize the importance of a certain phenomenon, that's unfortunate. But if you realize the importance and continue to pursue some mechanism underlying the phenomenon, it could be a very significant finding. The science is just like it. This is one of a comparison between the orbit trap and the power, the high field trap, and also general. This is another magnitude mode, the field, the frontier detector. This is depend on the physics of mass analyzer should use which kind of top. Like, well, if we do the metabolite usually use use the top. Either ESI or HCI. And the other usually can use for the molecule structure determination. That's what we have to use. Because now, when we determine the chemical structure, many often we depend on the chemical. It depends on the mass spectra. Because they very commonly applied. I don't know if it's because the mass spectra company did a good job to advertise their products, or just because their products really good to meet people. No other mice probably they don't have other options. No options, only gets those. But actually later on when the LC and TCMS have a lot of limitations, but they can do the very excellent job. For example, when we do the many fragrance, plant fragrance, our flower, our fruit, our tea plants, very extensive fragrance, so you can use the GCMS much more convenient because the library usually established according to the one is according to mass spectra mission, another according to the retention time. And those are basically now so far established the library relatively accurate and convenient, much more convenient than mass spectra. Mass spectra every time when it's done, unless you have like a phospholytic, the professor set up very good template. They use their own system. Every month, they try to adjust to normalize once or twice to make sure every time their template can be used, because the template already is like AI to define as long as the peak is within the how many minutes retention time, and what's the iron peaks, what the iron size, what's the value, they can determine this compound either the PC or PE or PI. So use this template, they can quickly reach the high throughput or high throughput out. You definitely, you just one sample example, you can generate so many different molecules because like phospholipid, the variation only is PC, the phospholipid, the phosphatile choline. The variation can be huge. For example, the two chains, fatty acid chain can be 18.1, 18.2, 18.3. Right. The second, if you have different combination, then you change that. And also, so this is the trap and Q-top. The difference is too small. But anyway, we already described, I explained the top is usually for accurate molecular profiling quantitative, and those are other ion trap our photo just for the structure determination. A Q-top device keep our ultra high resolution. Just for 400 can get reach that high. But recently, when they increase the energy, the detection of the molecule can be reached like more than 1,000. So this already reached very powerful. The detection scale, much larger, much powerful. So the new ESA, the top mass and mass, they also couple mass and mass for larger molecule to more accurately determine the property of chemical structure. They couple mass and mass. So this use capital mass to further determine the structure. So it makes the metabolomics analysis tool more powerful. This is for the orbit trap of the Q-top. The difference. Yeah, from this one, from this figure, you also can see the number of the ion for the detection. The top is much powerful. It's much powerful than the other orbit trap or quadro. It's much powerful than. That's why we normally use the Q-top. Then because when we do the mass, doesn't matter with the TC or else the mass factor. The most important limit is in the separation. The separation part is very important. UPLC and HPLC, the difference is like this two-dimensional the liquid chromatography, 2D LC. If you have two columns put together, you have much more powerful separation. Like this, the previous one peak, but very close. But then later on, when you couple the two columns, you can separate very well. Yeah, separation is very, very important. HPLC usually, we saw, normally HPLC, we saw this peak, this is the ETCT, but actually not. In LCMS, you can find the problem. Many LCMS, you couldn't separate C or DC, C or TC, DC. Or sometimes ETCG and TCG, they have similar molecular weight. They only have a different configuration. So you use the 2D LC can reach the separation. Then further, for the mass spectral analysis, can reach much higher resolution and also the accuracy. Because in one peak, if you hit after ionization in the electric field, this is one of example for the two D LC. One is the reverse phase DLC. For this word is reverse field. Usually we use the reverse. We barely now use any then second, use the cation exchange. Then you use the second field, second phase. You can the first phase you can only separate this. The second phase you can separate, just like a 2D protein gel. You can separate the protein spots very very well. So at least can reach another threshold, right? Segment or even 10 threshold. This is a gas spectrometry. What I think the simple, mainly the mass spectral, the quarter-of mass spectral analyzer. Here is the quarter-of mass spectral analyzer. And not the LC have 2 top, but the GC only have this 2 top. Then ionization, they usually use electron ionization. Very simple, but very powerful. Why? Because they generate a library. You can very convenient. Every people can generate it. Basically, do the analysis by themselves. And those most likely is accurate. But for the LCMS, many data is generated. If not one by one peak checking, there's a lot of mistake. But the GCMS, so far people think much better. Probably because they analyze the molecule relatively small. So the people call it GCMS is the sophisticated instrument technique that produce and separate and detect ion in the gas phase. The gas phase is definitely, because here you need to have a column helium or other nitrogen. Sometimes people use nitrogen, but most often we use helium to carry out the sample and do the ionization. Through the column separation, the ionization then gets into the quarter-of mass spectral analyzer in the electric field. It goes, then hits the detector. This actually we have a carrier gas, the helium common, then inject sample quickly in the vacuum with high temperature, because this injection is liquid extract. Doesn't matter you use hexane or other very easy evaporated solvents. You can quickly get this. And then through the very long the gas chromatograph, this usually use the capillary column, very long, then to reach the mass spectrometry. we trust this is because these use the causal method, the filter. So we only detect the certain compound with very strong signal, so that we can accurately get the mass spectrum. So sometimes reduce the noise. Okay, so that's why we use the gas spectrum. Usually people accept we just use the machine, the library we already built. And if you want to purchase our library, that's fine. Just follow their protocol, do the sample analyzing, then couple it to their library. You can, for example, I do like a spawning. I do spawning even use the TCMS. That's fine. I can very easily separate the monoterpene and sesquiterpene and also diterpene because the molecule is slowly increased. Ion charges are very similar. Then you can see the diterpene or some stereo in later, come out later because the molecule is very large. But those detection or finally give you the match, the better match suggest the molecule is very good. Usually you can define by later use the other machine. If you confirm this, that's good. So you just use this. You either can quantify, you can use this TCMS to quantify. Okay. We use this analyzer oil components and also analyze the gas and like chemical wax later I will talk this similar. So for the sample preparation here, because in morning we already talked about the different analysis. Some of them the organic acid also can be analyzed with the gas chromatography. But they use different, after you purify it, dissolve the organic acid. Organic acid usually you from the source, when culture medium or the primary extractor are many salt. So you have to dissolve. Use the HD10 column and try to dissolve. After dissolve, then you can do the derivatization reaction. Methylation or use the silicalization. There's some special chemical for the particular wax analysis. We usually use two chemicals from Sigma. One is the MAFS or something. I think cotton fluoride is something that's very easy to gasify, and also very reactive to modify all those the chemical wax because components are very complicated. Not only coating the fatty acid, broken down smaller, like aldehyde or ester or alcohol or acid, all those types can be analyzed very good. And also glycerol, you can see that. It's so very powerful. The sample preparation for the fatty acid, the sugar also can use TCMS to analyze. So people develop a lot of methods try to use the TC much more convenient and portable. The mass analyzer is the heart of mass spectrometry. This section separates the ion. Separation is very important. However, he used the causal. So the filtration happens. So some ion will be deleted, but some the very strong signal will be detected. So usually those the pass-by signal, detected signal, strong enough to get the reliable, the mass spectrum data. That's why you see either in space or in time, the separation, ion separation in both space and because they have a filter, and also in time, according to max to the charge ratio, because the flying time in the field, electric field is different. So after ion was separated, they are detected and the signal transfer to the data system. So I think the high vacuum will minimize the ion molecule reaction, scattering, and neutralization of ions. The GCMS, actually, I think if one new place want to set up, usually first set up GCMS. My experience is I see many places you will go by try to do the mass spectral analysis or try to train people use the GCMS first. The one side can analyze many important molecules, another side is less cheap, less expensive. Yeah, less expensive. But recently, people realize that many labs try to buy GC rather than buy LCMS. They try to develop a new method adapted to the GCMS. So the GCMS price increase a lot. I don't know what, but just there's some companies and so the electron ionization is the most common ionization technique used for the gas spectrum. Work very well for the most gas phase molecule. The limitation is also the mass spectra are very reproducible and widely used for spectral library, and easy cause the extra fragmentation, so that molecule you not observe is for many compounds. Yeah, that's also another reason is because the core drop itself have selectivity, have filtration to remove some more signals. So that's why this have a kind of a penalty. You get so many good advantages, but you also have penalty. This is not very satisfying. It seems always like this. So I still think that for initial metabolite profiling, the GCMS is a very nice machine to get into and first and adapt to it. And after people are familiar with the metabolite profiling or gas, you can further go to LCMS because that's very expensive. This is one word. Couldn't see very clear now. Well, this is one example. We did the tea leaf cuticle wax. Cuticle wax change in the tea leaf. Just add water. I said before, we just put the leaf. For the detached leaf in color form quickly, like in 30 seconds or not more than 1 minute. Then collect concentrate the chloroform, then you will get those the lipid compound, and sometimes you can get plasma. So basically, in younger, you will see a lot of fatty acid. Usually, from here, I continue to see the paper published in the Food Research International. Initially, we just try to understand when people making the tea, one most common observed thing, even those He already said, use the apical bar of young, very young leaves. You cannot get very good tea. However, it's a mature leaf or past the stem, you can get better tea. Black tea. The reason is that they believe that something probably in leaf. But in leaf we detect is a very different situation. In leaf catching, seeing those major tea, bioactive and caffeine, all the content like this, this is caffeine. In second leaf reaches the highest level. In apical bar, they also that's not very tall, but in this one also high. When leaf get older, the caffeine content quickly decreases. So this situation also true to a different caffeine, TCT, or those caffeine, also to the non-protein amino acid theanine. We know the theanine is very helpful, very healthy for promotion in this field. So this the trend we don't know why this can correlate with the tea, the processing. Very experienced tea processing said, okay, we use to mature it, we can make better, best the black tea. The best black tea. But you cannot use the young, very young leaves to make best black tea. So you analyze the water cause compound. You also is the opposite. That's why we try to understand why. Is there any extra reason or other reason? So we know that during leaf development, sometimes those compounds increase. And also, we know different varieties. We profile those alcohol. Those after the analyze, this just we can observe. But the other profile data, the much more data come out from GCMS. It's the alcohol, acid, ester, the candy, alkaline, and the sweetener, and also tritopene, sesquiterpene, and alkaloid. Alkaloid is the caffeine, and also sugar alkaloid, the glycerol. So there's so many compounds from carbon 3 to carbon 36. Very large molecule can be profiled with the GCMS. So very powerful. So our conclusion is that with the different variety, we choose this. Actually, initially we detected, we examined like 6 and 8 or to 10 different varieties. Finally, we choose the 4, and some the high particular wax loading means the leaf have more wax, particular wax. But the other 2 varieties have less, very thin, the particular wax. And we compare this metabolite finding after making into a tea. This is the initial. We did not prefer. This is after processing to tea. And we found that the calculated wax loading on the leaf more, then they were released more those volatile compound after processing. In the processing, we don't rolling or something, just like the white tea, just put there. They naturally put there to generate the white tea. So we don't processing, so our profiling only from the leaf. We're not artificially broken down so that information is obvious. The more particle exploding, then you observe the more stronger the fragrance smell. That means the tea quality is better. If you judge the tea through the smell.
Should we take 10 minutes break? If you
Okay.
because of the
Yeah.
break.
Okay. Yeah, now we can.
Is a break?
Yeah.
So we have the 10 minute break and we have the tea as a snacks for you for the dinner for evening, and after that we assemble here for next 45 minutes.
Okay.
Then it will be the class will be up to the 4:30 maximum because 4 hours we need to complete. So he's coming. You can take the tea from the here. You can go.
Yeah, this is last. We just finished the TCMS. Explained how TCMS different from LCMS. But basically, all of them are coupled to MS. But the TCMS have more options in interface. They use the ESI, or use the HCI, or use other type of interface to finish the ionization. Then the abstraction also in the detector part, and then also in the detector part, or in the mass separation part. In mass separation part, we have different of control, our executive arbitrary. All those make difference. Final detector or the mass spectral matrix. We show there are different advantages. So we keep in mind for those options. So which one is really work for you or good for you? It depends on your target and your purpose. For non-targeted, many people use the two-top. Just one time we can amplify and detect as more metabolite as possible. So those for non-targeted. But for targeted, and you want to look in details, what's the molecule? Particularly in some part you will be interested in those large molecule. So probably you use the Q executive. Even separation not so good. However, Q executive can even from mass spectra, you couldn't see there's so many ions just put together. However, they give the detailed information each peak, even this peak, included many metabolites. They all can broken down into the different ion fragments and to the detection. Of course, some of them you couldn't detect because the filtration, we filter some down. So those are, I think, keep in mind. Then that's it. For GCMS, I prefer to use the TCMS because their library they also use the control, have a powerful detector and accurate determination, structure determination, preference. So those I think very good, and also they have a very reliable, reproductive library. And recently people use even develop the model method to derivatizing more liquid, more larger molecules. Normally people use it for LCMS, but now you can use it for GCMS. So that's quite good. You have a new library you purchase from the machine producer or the software, analyzing software, the provider, so you can get very good analysis. Just do it by itself. Not like even, I think for China, in China, even we have so many machines like our own university have so many machines, but left students those by themselves to analyze pick one by one. No, you cannot trust because everyone have limits, the patience and durability, reliability. They cannot sit there for a long time to stand. You don't trust. Sometimes they just mess up. And so in that case, if not professional, LCMS analyze the data. Many often have a lot of mistakes. I have to tell you, this is true. Unless some people say, okay, we from large amounts of LCMS analysis or metabolomic analysis, we focus on some group of metabolite. Okay, this make sense. You only can focus on some point, some group of metabolite. Then you track this metabolite in wild rice to cultivars to land races. You can track how those metabolites originate from the wild rice or from enhanced selected in cultivar. And those stories are good, right? You have to focus on some. If some paper just tell about hours of metabolite, like this case, I have to say, the trustability drastically decreased. Nobody can accurately analyze all of those. They're almost impossible. However, for GCMS, they also present a bunch of very high-profile data, but many often those data kind of reliable. I hope to tell you. Yeah. If they also there's some calculation multiplied like a mass spec, MS, molecular weight, and also mass spectral ion, and also retention time. If they times those together, make an index, that's more reliable. For a reason, you can think about why this index, there are some lab established international available, accessible data. Just use the retention time, molecular weight, and other to the index. Different machine, different GCMS machines come out, then the index is very similar. That means reliability. So I think this is very good for the GCMS. Even though myself use LCMS much more than the GCMS, but we only focus on the certain part of it. We don't do the large scale data. We don't have the ability, we don't have the patience to do the reliable data processing of compound identification. If we don't reliable, then we just don't publish it. Yeah, I think this is the. So from this point review, I think we have some time. I will just quickly tell about the biological part. Biological part is much more important. From everyone, we study the physiology of from plant or from animal, from different. You always consider your metabolite in certain circumstances, right? For example, you do the metabolite profiling, you're concerned about the yield. You want to care about the photosynthesis, this metabolite. So from a large scale non-target profile, you only take out all those related, all those compounds involved in the carbon circle. In this cycle, you take out the compound to check if this make sense. The level or the very standardized or under different treatment. If you see, for example, in light or in some other situation that interfere with the photosynthesis, and you see those change dramatically. That makes sense, right? The metabolite always you solve the issue like this. In certain, in certain circumstances, that all interfere with photosynthesis, or for example in dark or sometime. So no photosynthesis. Of course, the carbon cycle is become weak, and then respiration increase. So in that case, what's happened? There are some reference people already studied as light in daytime sunlight, regular sunlight. What's the carbon cycle, the metabolite involving the change, how they change. There's some reference, and also from the RNA-seq, you can see the corresponding enzyme, how they change. Corresponding to the same sample, you do the RNA analysis, one part you do the metabolite profiling. The target profiling only focus on this, only our focus on the starch synthesis or glycolysis. So you can realize that, oh, that's really many of those make sense. That's good. That's how metabolite profiling really give you very deep, very personal impression of this really amazing, the data fit, the common sense. Photosynthesis in daytime very good. You can see those carbon circle, the carbon 5 or carbon 3 hydrate, metabolite, the cumulative pattern. And also at night, you probably see this, right? And daytime and night is very different. You can see those citric acid cycle change because those are all glycolysis become active, generate the energy for plant growth, and night grass growth. Of course, in summer, in the growth time period, there were more obvious. So from this point, the glycolysis and generates the citric cycle in the mitochondria or in the cycle, and this also in the this glycolysis in the cytosol provide the energy and also substance to the later fatty acid biosynthesis or protein biosynthesis. There's some transferrin factor also. Before, my experience tell me there's some like a RICO1, the transferrin factor T2, transferrin factor specifically regulates the PEC or the PFK to catalyze sucrose broken down, generates a lot of downside the organic acid facilitated for the lipid biosynthesis here. The generates the citric acid, acid CoA to generate to facilitate the fatty acid, the biosynthesis. So some transferrin factor just target on those very primary, very core part of metabolite pathway, metabolic pathway to regulate the direction, the common down participation, the carbon flex flow to the fatty acid biosynthesis, or I remember in Malaysia or in India, I think one scientist published a paper side, except the soybean. Soybean is not just oil-seed plant, but the palm seed also. The palm oil, some palm is like accumulate much more starch, but some parts accumulate much more oil. So the oil content and starch content, these are two different varieties. But I have a very dramatic metabolite accumulation pattern. Finally, after they did the metabolite, meanwhile they did RNA-seq, and they found compared what's the key transcription factor regulates the fatty acid biosynthesis chain, and they found the wrinkle one. And indeed, they found that the starch accumulation in the palm actually have the wrinkle one, the gene expression effect, the many gene structure. I don't remember this, like in intron, like a promoter mutation or some other reason causes the gene become defective. So they couldn't direct the carbon to oil biosynthesis, so their oil level is dramatically reduced. However, their starch accumulated. Of course, there are other regulator also regulates the wrinkle one, like in normal plants, while some plants, where most plants the seeds don't accumulate too much oil. Yeah, that's because there are other factors to control the wrinkle one expression. But anyway, those not only the carbon addition, but also the carbon nitrogen simulates the partition between the salts and zinc, like roots or leaves. That's also we consider can largely explain the same type of like carbon-3 plant, the yield, low or high. Yeah, there's some very tight correlation. I think this part of the problem is just the regular when we connected to this morning section about the fatty acid, glycoside, or something. All those is what we can profile, citric acid, all those. But the one thing is about the fatty acid is before I worked on fatty acid like soybean, so manual work. So I'm very interested in this. Even now, when we work on the tea plant, there are another camellia family plant. They call the Camellia oleifera. Camellia oleifera in China, they produce the larger seeds. The seeds, the oil content is very high. They also selected by farmer to produce the oil as vegetable oil. So those oil contain the high level of oleic acid. Yeah, this is oleic acid. So like this, just like olive oil. Like oleic acid can reach like 85%. The whole vegetable oil, the fatty acid, and the oleic fatty acid is very high. So similar to olive oil. That's why in China we call it the Eastern olive oil, the local oil. So we found that they also have similar problem with fat. Fat 1 and fat 2. Those cannot the only one double. They can into the linoleic acid and also linolenic acid. Those linolenic acid we know is very for healthy is good, but for the shelf life for the products, food products in shelf very easy oxidized. That's why this one in the oil processing, normally people, I know India have banned the trans fat. Do you? You have banned the trans fat. In food, you don't have allow the trans fat. Huh? Okay, let me. Trans fat is when the food produce oil, produce those vegetable oil, the salad oil or whatever. They usually put the oil in tank, then put the hydrogen, then also have catalyte, metal catalytic, then put there together to add the hydrogen. When you add the hydrogen, the double bond, those double bonds become saturated. Yeah, they add the hydrogen. However, add the hydrogen, they thought, okay, we generate the saturated, heavily saturated fatty acid. But actually, later on, people detect those fatty acids artificially as the hydrogen bonds. No, they are called trans fat. Naturally, this is cis fat. This should be cis, naturally cis. But artificially as trans fat. And nutrition medicine study from the trans fat have many bad negative influence. Diabetes, of obesity, and also other metabolic disease caused by trans fat. That's why Monsanto, very earlier, they developed omega-9 canola and organic high oleic acid soybean. They just try to produce high oleic acid. They get rid of those because the government in 10 years, 2010 or 2008, European Union banned all trans fat. You cannot see of any food have trans fat because they believe that's already fact. Even Monsanto or those are big agriculture companies already realize this problem. They start to produce this one. Oleic acid rich soybean and canola with rapeseed oil. So they already this one actually have a patent conflict with DuPont. Monsanto and DuPont fight together for the high oleic acid soybean. Finally, DuPont lost they just I think paid how many money to DuPont. So the high oleic fatty acid soybean will be on the market very soon because they already left the Brazil and Argentina, the soybean produce the high oleic acid soybean, and also together with increased several trees like the yield increase and also another anti-herbicide. Yeah, you usually use the phosphate selection, but finally add another 2,4-D or I guess this largely in the D2. So this actually very big achievement. We understand, okay, through the nutritional fatty acid and metabolite profiling, they finally bring out the high oleic acid variety, not only soybean but also rapeseed. All depend on the metabolite profiling. You screen large amounts of soybean varieties. Finally found some variety have higher, like 75 or 65. And another have, when you combine those together, that time they don't know which part of mutation. they combine together, find they are different. Those are different, different gene mutation. They have an additive effect. Combine together, separate those two genes, then can reach the 85% of oleic, oleic acid accumulation. So that's reached their goal. Finally, they purchased this patent from a professor in University of Columbia, in University of Missouri, in Columbia. Yeah, Missouri. Yeah, it's a very, encouraging story. Of course, after you purchase it, then we just bury this patent. We don't use it because they already generate the transgenic soil. So we just give up. So I mean, it's a very, very exciting story. The two companies, DuPont and Monsanto, fight to fire up the high oleic acid patent. Yeah, another part is also omega-3 fatty acid biosynthesis for the metabolic engineering. This part also people work on the grape seeds. I guess in the future we will get very good EPA or DHA. Those highly expected omega-3 fatty acid benefits for the kids in the brain, but also for the adults too. So this pathway is developed right now. We're working on that. I think our metabolite, the crop yield, nutrition quality, our QTL trait fine determination, the compound together usually make a very significant work or achievement if you work properly. Because right now, many countries, just like when we talk horticulture crop, the nutrition, quality nutrition, like carotenoids, our pepper, the spicy taste, our sweet, orange sweet taste, so on like tea plant, we expect to increase the theanine, the non-protein fatty acid, the non-protein amino acid, and also caffeine or other, try to reduce the caffeine, reduce the negative impact of this caffeine on sleeping or other. So I think all those can have a very significant. I just try to use this to inspire everyone. Yeah, that's how the metabolite widely used. Yeah, for secondary metabolite, the main is flavonoids, terpenoids, alkaloids, and other phenolics, the lignin. Phenolics, many of them are actually related to lignin biosynthesis. But the lignin is another larger molecule, sugar, coumarin, flavonoids. I think you know all those metabolites. Flavonoids, too many flavonoids. The structure have some all those natural products from specifically, for example, this one is in one tree, right? In one tree area, how you can accumulate. Camphophora or medicine and curare. There's some Chinese name because we also
have any question or communication with your colleagues, so I don't need that.
So the house is open for the discussion for the next 15 minutes. You can ask the question relevant to metabolomics. You can ask the question related to the topic we are covering here. You can ask all the question relevant to whatever you have in your mind related to the research. And yes, if we have some spare time, so we would like to take a few questions regarding Chinese research also, but those will be the last one.
Yeah,
We can discuss about the research. So yes, ask whatever in your mind. Monica? Monica? आप खड़ा करेंगे कुछ? अभिजीत? कहाँ है? Yeah, अभिजीत. Yes, it is a discussion part. You can ask whatever in your minds also. How you are coming from the outside. They can ask you what type of research they are doing and how, what they do their work much better. So what is your inputs? So definitely you or me will give the inputs on that topic. Yes, there is a gap in this course. The ice breaking was not done in the morning. And even I realized that I was felt to break the ice breaking. Yes, but however, whatever in your mind you can ask, we will give you more inputs for your research. So anyone who would like to ask any question? They need to some space, more space to open up the ears.
Yeah.
That's the reason. So yeah, we would like to finish now. So thank you, Professor Jain, for this presentation. Yes. Yeah, you ask. Ask whatever. Give me this. Ask.
Sir, I'm a master's student here at Central University of Punjab. My current dissertation topic is based on nanoparticle synthesis and its characterization. Sir, the bioactive compounds in algae have been used to synthesize the nanoparticles for biomedical applications. So, my question is, if you are performing GCMS and LCMS, how can it be used to make it more pure, and how will it affect the quality of the nanoparticle?
Using the onion extract, the purified onion.
I will give the answer of it. It is working on the nanoparticle and you would like to use these GCMS in the LCMS for his work actually. So, yes, Abhijit, please sit down. For nanoparticles, you have to use little bit different type of the techniques. That is the AFM, that is the SAM, and that is the FTIR. GCMS and LCMS has the different quantification concepts, and basically these are the based upon the chromatography. In the chromatography, you can find out which metabolites when bind in some nanoparticle is a little bit later, which come earlier. So that type of the things you can identify how these nanoparticles are bind in the different targeted metabolites when you fuse those metabolites with any in the drugs form or in the form of some fertilizer or something like that. How they are going inside, how they are working with the another metabolites, so how they bind up, what is their binding specificity and their releasing specificity also. So for those that first of all you have to characterize the nanoparticles, and for the nanoparticle you have to use the different techniques. First of all the preparation of the nanoparticle, the second is the characterization of the nanoparticle, and third you have to use some model in which you can use those nanoparticles. LCMS and the GCMS basically they have two major differences. GCMS is used for the volatile chemicals. of the carbon atoms. Second is the number of the ring structures. So more ring structures, so definitely they are some secondary metabolites, we are related to them. More carbon number, carbon chains, so you will identify so many array diversity in the fatty acid. Remember, fatty acid is the most diverse metabolites in our living status. So for that, GCMS is using the techniques in which column has a very big size. Column is around 25 meters, about 50 meters also column that we are using. But in the LC column, UDLC and the LC, they are used like in chromatography, and you have to do some detection. They are using for the separation of the metabolites. So whatever the metabolites you need to separate, you need a technique. LC and the GC is doing only for the separation of the metabolites from a collection of the metabolites. Analyze. We are using a word that is the analyze, not the metabolites, because we have to analyze the component. So now it's okay. We separate all the analytes which are present in the mixture. Then there is another concept that will come out. How we quantify those metabolites. So for the quantification of the purpose is majorly there are two different techniques. First is the mass spectroscopy and second is the NMR. So mass spectroscopy is usually based upon some concepts, so I will cover that portion also. So when we integrate these two things, first is the chromatography and second is the mass spectroscopy. So now it seems to be give the answer of our question. So first part is separate the different metabolites. Second part, at least identify those metabolites on the basis of the charge that is present on that particular thing, and this charged particles behave differently in the different environments. So when they are behaving different in the different environments, we interact those information on the basis of those information. We differentiate the different metabolites in the different categories also, and we can identify what is the exact mass of that metabolite, charge of that metabolite. On the basis of that we can do. But we have to use some reference components also, some internal controls, some quality controls also, and sometimes we have to use the previous data that was reported by someone on the same component. So those reference metabolites spectra help us to our lab or to identify which is a new metabolite in the new situation, which is a metabolite which was not previously present in that particular solvent extraction. Why? Now we are able to identify that metabolite. So there are many parameters, solvent extraction starting. I will discuss about the metabolomics, but the most important that I can understand in metabolites, every step
Hall. First lecture by Professor Jain,